श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा। शुक्लां ब्रह्मविचारसारपरमाम् आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदां जाड्यान्धकारापहाम्। हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्।
Good morning, everyone. I welcome you all to the final day of our very exciting 5-day online workshop on overview of thesis from inception to completion. We had 2 sessions yesterday. Dr. Malati, Professor Department in Oral Pathology from Sri Ramachandra Dental College, Chennai, threw light on ethical aspect of research. And I'm sure all of us now have a clarity of this important aspect. The second session was by Dr. Puneet Batra, sir, Director PG Studies at Manav Rashna Dental College, and sir gave an overview of plagiarism. Both these aspects hold a lot of importance in research, and I hope that all of us have imbibed all the tips given by our speakers. I would like to take this opportunity to introduce our speakers and chairpersons for today's session. We have with us Dr. P. Rajashree, ma'am, Assistant Professor, Center of Advanced Studies in Crystallography and Biophysics, University of Madras, Chennai. Ma'am will be talking about evaluation tools in research. Along with that, we have Dr. Suneel Kumar Chinni, sir, Professor in Department of Conservative Dentistry and Endodontics at Narayana Dental College and Hospital. I would like to take this opportunity to invite our chairpersons for the sessions today. Dr. Ajay Logani, sir, Professor and Head Department of Conservative Dentistry and Endodontics at AIIMS, New Delhi, and Dr. K.C. Ponappa, sir, Principal at Coorg Institute of Dental Sciences. We thank you for your gracious presence here today. Without any further ado, I would like to request Dr. Ajay, sir, and Dr. Ponappa, sir, to kindly carry forward today's session.
Okay. Thank you very much, lady, and we're very happy to be here. First, I must congratulate Professor Arundeep Singh, Professor Mahalakshmi Madam, and Professor Dax for having organized this online event. I think they've taken the very good clue that these are all the positive outcomes of the COVID pandemic, which we had in the last 2 years. They made full use of, and specifically Professor Arundeep, I saw that during these 2 years, he made sure that the academic platform was on, may it be on the online version, but it was on. And that is what I think is the message, is that even in dire circumstances, something positive would come out. And I think this workshop is a positive outcome of this, a very much needed workshop for the first year postgraduates. And the way the numbers are going up on Zoom, I can really see that the last 4 days have been really, really successful. And I think it's been useful for everybody. Today is the last day, but an important day. We have 2 speakers here who are going to be speaking on extremely important topics. So I have the opportunity of introducing them to you all. Please, Dr. Rajashree is the first speaker, and she's an assistant professor at the Madras University. She is a PhD, and she also has a fellowship in the CSIR UGC National Eligibility Test Fellowship. And what I see from her biodata is that she has many projects, extramural projects. So I'm sure, because I do realize how difficult is it to get extramural projects, but she has 7 of them and 2 patents. So not only she's doing some good funded research, but she's translating them into reality and getting patents out of it. So I'm sure the message she has from her topic is going to be very, very useful. And we are more than eager to hear her. Thank you very much. Let's start, please. Thank you. Madam, could you now
Yes.
start?
Thank you, sir. Thank you for that kind introduction. So before starting the session, I would like to thank the organizers for giving me this opportunity. The SRM Dental College and Manav Rachna has come up with a very good program starting from what research and how to go about it, which even I didn't have the opportunity when I was doing my doctoral research. So I think the quality of research will improve at the student postgraduate level itself. And let me share my screen and start the presentation. So are you all able to see the presentation?
Yes, madam.
Yes,
Just,
yes, you can see.
Just
yeah.
Yeah.
So yeah. So evaluation tools in research. So if we see what evaluation tools, generally if you go and Google evaluation tools, it will show a lot of statistical methods and models, how a research can be evaluated. What are the way by which you can assess the quality of a research. But today we are not going to see that. Today I'm going to walk you through the different tools, especially biomedical tools, by which you can assess various parameters for different types of biomedical research. So before that, I just want you to, already you must have been given enough about what biomedical research is, but to summarize that, to recap all these 4 days, let's just see what it is for actually. So it is for knowing the cause, prevalence, and severity of a disease, and to develop diagnostic, therapeutics, and preventive measures for a disease. Now, this cannot be done by a particular individual or a particular group of people. It has to be done by several groups across the world, or across various research institutes. So there are various stages and phases of biomedical research. So what phase you are going to choose, that will define the tools which you will be using. So that's, you must know what stage of biomedical research you are going to be selecting. So how to do that? So usually biomedical research is not a very individualistic science. It is an integrated science, as today they say interdisciplinary, cross-disciplinary. So science, and especially biomedical research, is cross-disciplinary. So we have to use chemical, physical, mathematical, and computational approaches as tools to analyze our research in biomedicine. So where to do this? You can, as I told you earlier, you can do at any stage. And that can, and which stage you're going to choose will determine whether it is going to be an in vitro study, or whether it is going to be an in vivo study using animal models, or you're going to collect data and use bioinformatics tools and machine learning to assess your questions, or is it going to be epidemiological or clinical trials? And I have had the experience of working in dental college, SRM Dental College, for 2 years. And I have seen most of them, they design studies towards clinical trials. I feel you should also move to basic research and applied research. So let us see what are the types and stages in biomedical research. So I have been telling you there are various stages. So you must have known from the first day itself that medical research are of 2 types, right? Primary research and secondary research. So I'm not going to go into that, but we need to know
Rajashree, may I interrupt, please? Sorry.
Yes, ma'am. Yes,
Your
ma'am.
screen, the slides are not moving.
It is in the first slide?
Yes.
Yeah.
Oh my God. I am in the second.
Can you use can you stop sharing and share again?
Yeah, sure. Is it in the title slide or did it move?
The title slide, please.
Oh,
I thought that you were.
Even I thought it was just the intro going on.
No.
Hello?
now,
You'll have to screen share again, madam.
yeah. 1 minute. Screen share.
Ma'am, the same button again, share screen.
Yeah. I'm doing that, but
yeah, correct, ma'am. Coming, yes. Now put it on full screen. Yeah.
this.
Just moving now. Just now when we were doing it, we were doing that. Yeah. Yeah, correct.
Yeah.
Yes.
Now we are seeing it.
Yes.
So this was the first slide, which I was talking about.
You got this point, yeah.
Yeah. Good.
So
yeah,
now
correct. Perfect.
we are into the types and stages of biomedical research. So in the primary research, these are not the types, these are actually the stages: basic research, clinical research, and epidemiological research. So if you have to approach any research question in biomedical field, either you have to start in the basics, and if you have got a valid output from the basic research, which can be applied for clinical research, then you may have to develop into a product, validate it in vitro and preclinical research, then move to clinical research, and as a whole, you can see. And epidemiological research, you all know, if it is for knowing a cause or preventive strategy, or to know how a particular factor is affecting a population. So that we are not going to discuss much. I'll be focusing more on basic research today, as I'm a basic researcher myself. So if we see the basic research, that can be classified as two types: theoretical and applied. So most of the theoretical and again, you're not seeing, sorry.
No, we can see, madam. We can see, madam.
Oh, ma'am, going on, correct. Yeah.
If there is any problem, I'll get back to you, madam.
Yeah, sure. Okay. So if you say the theoretical part and mathematical development or physics, we use all the scientific. I'm sorry, I'm hearing an echo of myself.
Yes. So, madam, do you have any other system near you?
No, I don't have.
A mobile phone, madam, near you?
Yes.
So
now this would be okay.
Okay. So we use theoretical modeling, especially with theoretical physics or chemistry. We derive we have hypotheses, and we use mathematical modelings. Even in psychology, they use such kind of things. And in my department, specifically biophysics, we use a lot of theoretical modeling to derive structures and to see how things work. But it may not be true, but you get an idea out of it. Then there are also departments which develop sciences, which develop analytical procedures when we have to measure a particular bioanalyte. For example, simple glucose, we have a lot of techniques to measure it, starting from biochemical methods to now we have electrochemical and point of care devices, right? So all these are development analytical measurements which are developed in physics or chemistry or biology, biomedical laboratories, basic biomedical laboratories. And even imaging procedures, they are all developed and characterized in them. So you will be using all these for your, analyzing your application of drugs, or for diagnosis, for developing a diagnostic, or to see the mechanism of a disease or a particular material. So for this, you may be using all this. So if you see the applied, so whatever has been developed here, an analytical tool or a drug, that has to be applied, checked, So there we will be using the models. The model may be a cell-based model, which is going to be in vitro, an animal model, and the tools, that is how we will be analyzing, will depend on the technique which we will be subjecting. That depends on all other subjects, genetics and biochemistry, material development. So you in dentistry are using all these, right? So in clinical research, this you must be well aware of. So it will be an experimental. If there is going to be interventional studies, it will be also experimental, and you have different phases. I'm not going to touch upon that now. So basically, clinical research is used for to see how an intervention works, whether it is efficient or non-efficient, to see the toxicological profile of a particular intervention. So, and as usual for diagnostics and all other purposes. Even case series and single case reports is classified under clinical research. So in epidemiological research, you have again interventional study, which may be in a population. This is going to be a defined group of population, but here you will have a sample size which is going to be larger, and it will give a broad overview about a particular factor or a cause or prevalence, etc. So secondary research, I think the next lecture is going to be on that, which is based on the data of the primary research. You will be using it to do further analysis and synthesize a better interpretation. So that is what we do with meta-analysis. It is just the data which was subjected to further statistical analysis, and you derive better conclusions. And writing a review based on the original research articles. From that, we do systematic reviews. So I told you that you have different stages, and we saw what are the types of basic research. So in basic research, now that you know that, yeah, I'm going to do this, I'm going to identify a marker, or I'm going to analyze the efficiency of a material, or I want to validate an already existing marker. Now you have got a research question. Now, the next question which you should have is, what kind of model am I going to use to assess my research question? So please keep it in mind, this is only related to basic and applied research. I'm not talking hereafter, I'm not talking about clinical or epidemiological studies, because that is more pertaining to, more related to human-based or human observational data. So if we see the models which are existing, in vivo model is there, in vitro model is there, in silico model, ex situ, in situ, and ex vivo. So most of you must be already aware of these types. So let me just give you an overview of all the models for whoever doesn't know what each is. So in vitro model is a model which occurs, the word is derived from Latin. Vitro means glass, which we perform in a test tube or in a lab with controlled and standard laboratory conditions. So this is very valuable because it does not require animals, and you can easily get ethical approval, expedited ethical approval, and it's also cost effective. It does not involve a lot of cost. You buy a cell line, or you get a tissue model, or just to get the samples from different diseased persons, and then you can use it, manipulate it in the lab, and then see. But the problem is you can only assess a particular analyte or a factor with a controlled environment. You cannot get a holistic view about that condition. So that will be a problem with in vitro models. Otherwise, it will definitely give the effect of one particular analyte or one particular factor. You can do it. So the sample variation between the different duplications, when we are going to use primary cells or cell lines, the variation, if you're going to repeat it, is going to be minimal because it is from the same source. So, and the only problem is it is reductionistic approach. It will not give a holistic view of the research or about the conditions. That is the only issue. So the next model is ex vivo model, which biomedical science widely uses this kind of model. And it gives a more clear picture of the research problem, or it gives a better solution. So what they do here is they get the living cells or tissues or body fluids. Like in case of cancers, we get biopsies, right? So, and we get samples such as blood or urine sample, or in case of dentistry, you get GCF. So we take those samples, but we don't do any manipulation in the lab and just measure the analyte of interest. So that is called as ex vivo model. You know what is happened in the inside the organism that you are measuring outside. So ex vivo means taking the sample outside the body and measuring it. So you get a at least near reality of what is happening inside the organism. But the problem here it is an invasive method. Now these days everybody are moving towards non-invasive method, but that is one issue. And there will be a lot of sample to sample variation because each individual has a different genetic makeup and different lifestyle, and there'll be a lot of variation. So you need to have a high sample size. So you have to do a pilot study and then fix the sample size for a particular analyte which you're going to analyze. Or if not, if already some other group has done it and published, based on that data, you can calculate the data sample size and then fix your how many numbers you're going to do. But it reflects the exact status of biomolecules inside the organism for a given condition. It may be a disease condition, or when you have given a drug, how it is, what is happening to the drug over a period of time. This you will have accurate judgment about it. And it is also a substitute for using a whole animal, even though whole animal gives next we will see about it, but using an animal, sacrificing it for our purpose, it's still unethical. So there is another model, in situ and ex situ model, which is in situ means at the original position itself. When we talk about cellular research, we do mostly molecular and cell-based research. And I want to know how a particular DNA is manipulated, how far the expression is when I give a particular intervention or when I treat it with a particular drug. I want to see at the cellular level what is happening without disrupting the cell. I can go for such model in situ. So a perfect example of in situ is FISH procedure, which is fluorescent in situ hybridization. So what they do is they use a fluorescent probe and then they allow the cell to take the fluorescent probe. It will go and conjugate. Also, it will pass through the nuclear membrane and then bind in the DNA. I'm not going to disrupt it. Also, I get a clear picture of how far is a particular gene expressed. This is called as in situ, in the original position itself. I can do it by disrupting the cell and then isolating the DNA. That is also there, but that is not going to give a real picture. So this is another model, but this is somewhat the technique is not cost efficient, but it gives reliable data, reliable outcome will be there. This kind of procedure, FISH procedure, is used for certain diagnosis of certain cancers at the genetic level. It helps in identifying various mutations. So that is there. In situ model is there. Likewise, you have ex situ model, which means whatever there is inside the organism that is taken out and then it is given a particular standard condition and analyzed. You may wonder, is it not almost similar to your in vitro? But the difference is, in vitro, we just see the cellular or the biological tissue level. Here, the whole organ, for example, in case of here, you can see the liver is taken out. So here again, we use animal along with this model. Or it can be a organ. Now these days they have come up with organ bioprinting and all. So that kind of model, if it is available, it is all advanced techniques which I am presenting here. So the organ is taken out and then it is subjected to the condition which you want to study. So all other parameters will be kept standard, whichever analyte or condition you want to address, that alone will be modified. So that will give, and how the organ is going to respond to such a condition will be given. So this is way better than in vitro, where an individual cell or the tissue is given, because the whole organ, how it is going to act in concert with other cells, will give better picture. But it is like in between in vitro and animal model. But still, what is more important here is it will give the functions, particular functions of an organ of interest. So this is the model which everybody knows, in vivo model, the animal model. So in vivo models, we use, whenever there is a new drug developed or a new intervention which has not been tested and used in humans. So we want to know whether it is toxic or not. Before using it in humans, we test in animals. If there is toxicity, of course, we stop it. And not only that, it helps in development of easy disease models, because the small animals like rodents can be mutated, or it can be gene knockdown can be done, which will help in development of a particular disease model itself. Exactly, in cancers, we have a lot of mutated versions, and such kind of versions can be developed. It is possible. There is also downside. We see that it is similar to human physiology. There is to certain extent, not exactly, but still there is a downside also. One is there is ethical concern. Then the second one is, I think in 2015, Harvard University came up that 98% of animal-based study is not applicable or is not mimicked in humans. Why is this? The extrapolation which we do with statistics is not exactly matching when we do when we test it in humans actually. Why? Because there is always, there is difference, genetical difference, and it is also kept in standard conditions. It's dietary factors, and there are a lot of other physiological differences prevailing in these animal models. But yet they give some idea about whether it is, at least we can, now these days we can know toxicity. For that only, now these days, this animal model is being used. For basic research, we have moved on to like organ on chip, 3D printed organs. So such kind of alternatives have been developed now these days. So if we see animal model, you have in this picture, you can see that there are a lot of models. All these are employed. So which kind of animal model you are going to choose depends on, again, what you want to study. So for example, in case of bones, they usually go for, if it is like femur bone, they use mice model or rabbit. But if we have to see something or test something related to tooth structure or alveolar bone development in the oral cavity, so then mice may not be a relevant one, because it is very, the anatomy of the oral cavity in mice is very small. It is not easy to handle. In such a case, we should either go for rabbit, goat, or dogs. So you should only decide what your animal model is going to be if you are going to conduct an in vivo study. Okay. So here is a list of what animal models are used for which research. So cats are mostly used for ophthalmology and type 2 diabetes. It is not mostly, it is subjected. Fish is the one which is now these days used, especially zebra fish. It's very small, easy to maintain, easy to reproduce in the lab, very cost efficient also. So for drug screening assays, behavioral studies, even in your labs, you can use zebra fish, not for studying bone growth or related to dental structures, but other behavioral studies, if you are interested to particular thing, that can be done, and immune diseases related related research can be done in fish models. Then Drosophila melanogaster, fruit fly. This is usually used by basic researchers because it's easy to grow faster within a day and maintain. So mostly when we do genetic studies, molecular biology studies, and cell signaling, if we want to see a particular mechanism, identification of a molecule, for all these we use Drosophila melanogaster. And also for drug screening, they are using. So likewise, goat and sheep for bone tissue engineering, guinea pig for stem cell engineering, especially related to cardiovascular diseases and for pharmacological characterization. Horse, you know, it is used for development of anti-serum and anti-venom generation. So for whatever we have, the TT and all injections, it is mostly developed in horse. And the antibodies which we have, like polyclonal antibody and monoclonal antibody, they're all developed in rabbits and mice models. And usually in biochemists, immunologists, and all life science research, they rely on mice model because the genetics is 95% similar to that of humans and easy to breed. And the physiology is also almost similar to us. So as I said, it is not going to be exactly as human, but at least it gives an idea if you have to see as a holistic approach. In vitro will give, but it is only going to give the effect of a particular molecule or a factor in a standard laboratory condition. But in reality, that is not the case. There are a lot of factors and molecules influencing inside the body, right? So if we need to know that, in vivo is the best option. And similarly, wound healing studies, if you are developing a material for wound healing or developing a graft, pig, mice, and rabbit is again a good model. Okay. So I was talking about organ on a chip. So this is what is being used in almost all the advanced research institutes who are developing new drugs or interventions, diagnostics. They first check with organ on a chip. And if there is a better lead on the organ on a chip, probably they test for toxicity in animal models. Before whatever was developed, it was directly checked on in vivo models. So what is an organ on a chip? So you can see literally it is a chip, like a silicon chip, where using my two important factors, microfluidics and microfabrication, they replicate the key functions of living organs. So now these days, a whole set of organs interconnected. So here you will have the heart, nearby lungs. So such kind of whole human-based physiological arrangement itself is being developed on a chip. So potential research tool, it is there in the market for particular organs, like for heart you have, for intestine you have, but it is still very costlier. Probably in another 5 to 10 years, this will become a easy go for. Probably it will come down to some thousands of rupees in your future. We are waiting for that. When the additive manufacturing techniques become feasible, 3D printing and bioprinting becomes cheaper, this will also, the cost of this also will decrease. So there is a potential application of this in all the fields, not only in pharmaceutical and chemical applications, for studying mechanisms, everything, for diagnosing, for toxicity studies, for all people have used, and it has been reported in research articles. Okay. So why tissue models are used? You name it, those tissue models have been developed. Finally, this is the in silico model, the very favorite for our biophysics department. We do a lot of in silico work. So at the level of molecular level, and specifically at the atomic level, if you want to see what is happening, in silico model is the best go for, because you can
epithelial வந்து எப்படி கனெக்ட் ஆகும்னா பேசல் வழியா கனெக்ட் ஆகும்.
So if you are want to study at that level, then you have to go for in silico models. There are a lot of advantages with in silico models. It is faster. Say, for example, I identified a target protein for a particular disease. Let's say periodontitis. For that, I identify a target protein. But against that, I want to develop a drug. If I am going to see the literature and the database, there will be millions of molecules available. If I have to test each and everything, it will take decades, not only decades, my whole lifespan will go in that. But today, within a week, you can screen those molecules against your target protein. So we have lots of computational tools and software where you give the structure of the protein and then you dock it against those structural small molecules or phytochemicals, you name it, whichever compound. You have to have the structure of both, and there are automated versions are available. You just leave it for 1 week. If you have continuous, you need to have the continuous power supply for that. That's it. And a high supercomputer will help in doing the job even faster. So these are the two things. But one thing is, you cannot really rely on this totally. It will give you again a accurate. It will get you towards or close to the reality. It's not the real fact. Say, for example, I told you that there will be millions of lead compounds. From that, you can screen and get say hundreds of lead molecules. So from millions, you can filter out, but you cannot say that, yeah, this is it. You have to do wet lab work, experiments in the lab, either by in vitro assay or whatever models we saw. And then you have to validate that, yes, this compound is actually inhibiting or it is actually curing. So that has to be done. You cannot say with in silico assay, in silico, modeling itself, you cannot get to a point. So for drug prediction, as I told you, we use, and molecular docking studies is the one. We have a lot of tools like freewares are there, like AutoDock. Similarly, as we saw here in organ on chip, even toxicity studies can be done by in silico model. An example of that is a tool is there called as ToxCast, which will give you an idea at least. Yeah, I have got this lead compound, but I need to know how far it is going to be non-toxic. It may be effective, efficacy may be higher, but how far it is non-toxic, that can be predicted. Later on, like even clinical trials. Now these days, ISCT, a new terminology is coming up, where they do clinical trial also in radiology. So a virtual physiology is developed in silico, and then they subject it. Even drugs are being tested. So it is like, it's not exact, but gives you an idea. Okay. And we use virtual cells. In a cell, if I give a drug also, I don't know what is happening to that cell, whether it is a drug, whether it is going to this lysosome, or whether it is going to enter into the mitochondria, or it is directly going into the nucleus, how it is being taken up by the, what is the mode of uptake of the drug. So all these basic curious questions, mechanistic questions, can be, you can get an idea by using virtual cell. You can Google all this, you can see what it is, because I cannot go and explain you all this, but I'm giving you, I'm exposing you to all these versions. And the final one, which is coming up now these days, is systems biology tool. So I was telling you, holistic approach is what is better for biomedical research. That's why we do in vivo studies. Now, ethical issues are there when we use animal studies. So there is this tool called as, we have clinical trial as well as system biology tool. So either the whole tissue or organ can be modeled. So mathematical tools along with computational software tools are used, like MATLAB, and the virtual cells are combined together. Like you get the networking, how each the proteins are interacting within themselves inside the cells or organs or tissue, or even ecology based study, population based study can be done. So either you can observe or you can simulate. You can make an intervention in the tool itself and see, if I perturb this factor, what is happening. Yes, that can also be done. My slides up, yeah. So until now, I told you these are the models available for basic research. So clinical trial-based research is what, if you have 50 students who are doing MDS, at least 30 of them, I see that they do clinical trial-based research, academic clinical trials. So before that, what is more important in clinical trial is what, before starting your research, you need to register it. This is more important. If you're not going to register it before recruiting your first subject, you cannot publish it. So that is, it has become a mandate. So we have a trial registry in India. So there is international trial registry also, which is being formulated by WHO, and there are NIH clinical trial.gov is also there. But this is a part of it. Our India trials registry in India is a part of it. So if we are going to register it, it will also reflect in those databases. Why this is mandatory? Because we need to follow the ethical guidelines. The public has to be informed what is the research design that has been done, and whether it has been followed, and what was the outcome. That has to be informed to everybody. And we can also avoid duplication of research. So, the cost of research is higher, and more than that, the samples which we get from subjects, patients, is also valuable. So we cannot waste it by duplicating it unnecessarily. So this is what is about what I wanted to insist on, clinical based research. So there are various phases in clinical research. So laboratory research, which determines whether it is useful and safe, you can get some idea. And in the phase when if you find that yes, it is useful, it is effective, then what do? You have to do the phase 1, where we just take healthy volunteers, not the diseased persons. Healthy volunteers are taken, and we see how the disease, drug metabolism is, how the intervention is. If it is in case of drugs, I'm telling. If you find that it is non-toxic, then we go for testing the efficacy with higher sample size in patients. So mostly you may be within this phase 1 and phase 2. So know the mandates and the requirements for performing the phase 1 and phase 2 if you are going to plan a research on clinical trials. Yeah. So until now we saw how is what stage you can be. You have to decide upon that. Depending on your stage, you have to decide your model. And when you have decided your model and you have the research question, either it be a basic research or a clinical trial, you have to measure. That is what a science is all about, right? There you need to get the data, measure the analyte. What is the way of measurement? Yeah, you have qualitative research also. We are not talking about qualitative research, that's a different aspect, and it has got its own significance. So we are now going to talk about quantitative research, lab-based research. So bioanalytical research tools, there are lots of bioanalytical, there's battery of tools in one, each and everything. Basically, we can classify as separation technique, detection technique, and higher end sophisticated omics based studies. So if you are going to be interested, or if you are going to do some phytochemical based study and want to analyze whether it is effective, and you find that yes, this herb is effective, and the next step is what is the component which is rendering that effectiveness. To know that, you need to separate out the metabolites in the component. It may be anything. I just gave you an example of herb. It may be from sea algae or from animal source or anything. Even from bacteria, most of the antibiotics which we have today is from bacteria itself. So how do we do that? It is purely from chromatographic technique. So chromatographic techniques, again, a lot of techniques are available. Liquid chromatography, gas chromatography, HPTLC is high pressure thin layer chromatography. Then if we go for detection techniques, yeah, I have a molecule. So you might be doing the separation, that itself will be a research, or you can buy a pure compound, which has been reported as a molecule having an effect on this. For example, curcumin is there, which is isolated from your turmeric. Instead of isolating it, separating it and purifying it, that itself will take 1 year. So instead of that, you can buy a pure form version from vendors for different manufacturers, and you can do your interventional studies and see what is the effect. For example, say curcumin is effective against oral cancer. So what are the parameters you're going to measure? Either some of the biochemical parameter or expression of certain immunomarkers or cells, receptors on the tissue, right? So what are the techniques which you will use? It will be based on UV, that is spectroscopic methods mostly, or imaging methods. I have not mentioned here like microscopic techniques. Either you can be using normal light microscopy or fluorescent microscopy, or in advanced cases, they also use confocal microscopy. Now the latest is 4D microscopy, where we see the whole cell, live cell itself. We add the drug, we keep it in the incubator, and it is attached with a microscope, and in the computer, you can see what is happening to the drug if I add a fluorescent tagged drug to that. So that much of advancement is there. So now we will see what techniques we have. So if you want to analyze these markers or molecules, then biomolecules, you either go for spectroscopic method or fluorescence based fluorimetry, flow cytometry, which is based on again fluidics and laser. Okay, so NMR is, these are all UV, IR, NMR, infrared. NMR is nuclear magnetic resonance and mass spectroscopy. These are all spectroscopic method, and this is sufficient for detecting quite a lot of biomolecules. Yeah, so as I was stating, emphasizing holistic approach is what is required to get a clear picture of a disease or a intervention. This is what people are working throughout the world. They are working on omics based studies. whole of the genome, instead of studying a particular protein, instead of studying a particular gene, okay, instead of studying a particular molecule, glucose or carbohydrate, I want to see what are the molecules which has been disturbed in a disease, or in a, when I give a drug, or when I place a material. How do I do that? We have to go for omics. So the method is LCMS, is again mass spectrometry based, but it's a higher end equipment producing data within 2 days or 3 days. You will have so some gigabytes of information on all the molecules. And there are softwares which give you which has been expressed in higher quantity or which has been expressed in lower quantity. And what are the molecules present in that? If millions of molecules are there, say in saliva, if you are going to analyze the molecules, metabolomics, which means the metabolites present in the saliva, or the microorganism in the oral cavity, microbiome, metagenomic study is being done. So there are a lot of omic studies available. And again, if you want to see the genomics of a whole cell, whole thing, NGS, next generation sequencing, is the one. MALDI-TOF will give you proteomics. All the protein profile will be given. Microarray will give you the expressed gene when we have a particular disease or a intervention. But mind you, this is sophisticated and involves a lot of cost and needs high skill to perform. Yeah. So that was about, I gave you an overview of different types on for what we use it. Otherwise, the common bioassays which we use is the biochemical assays. For example, your protein measurements or your glucose measurements in the lab, OTD, which most of you must have performed in your first year lab practicals, right? That is from using those assays also you can do a wonderful research. See, I am exposing you to lot of advanced, sorry?
Discipline ke puri band baja karti hai.
So
Go ahead. Sorry.
okay. So there are a lot of advanced techniques now these days. But the quality and the standard of your research does not rely on what technique you are using. No, that depends on what research question you are putting forth and how you are addressing it. Exactly the methodology and things are, and how useful it is going to be. So these are the things please keep in mind, and how cost effective is. So if you can do some good research with simple assays itself, even that can be published in a high impact journals, and you can also develop patents based on that. Don't think that I need to do omics only, but I'm just giving you an idea on what are all the analytical methods available today. Okay, so enzyme-based assays are there, which again can measure, you can like your STOT, all your blood profile enzymes are there. That kind of assays like creatine kinase measurements. And this is all, you know, even though it is different approaches there, all these are based on, these three at least are based on spectrophotometry, visible calorimetry and visible spectrophotometry. Enzyme based and immunoreaction based array, say like ELISA, mostly perio department and oral cancer, oral pathology and oral medicine, they use a lot of this. Even other departments can use, but based on their interest, I'm just telling you. Fluorescence based assay like flow cytometry and fluorimetry are available. Cell based assay, if you have to know the cytotoxicity of a material, dental material, or if you have to if you have a new drug intervention, so before moving to animal model, it's better to check with in vitro. So the test used is MTT assay. There is a drug, there is a compound called as MTT. It's a very big name. I'm not elaborating that. It undergoes an oxido reaction inside the cell. So once the cell is live, it will not, it undergoes a change in color. When the cell dies, the This is a dye actually. The dye will turn pinkish color. The more pinkish, it means the cell is dying, cells are dying. So that is a way by which you can measure the cytotoxicity. LDH assay is also there, lactose dehydrogenase assay, because this will be released from dead cells. The higher the LDH activity, you can know that there is more cells dying. So there are a lot of assays for measuring the cytotoxicity itself. Flow-based assays. This is only a few list I'm giving. So you can study based on these. I hope you're all making a note of this, because this is not the end of all. I am just touching upon the tip of the iceberg, I can say. There are lots. Okay. So nucleic acid based assay, now even common man is aware of the different nucleic acid based assay, especially with the RT-PCR due to COVID. So PCR, RT-PCR, gene sequencing, and RFLP. RFLP and gene sequencing can be used for polymorphism study also. I have seen some of the departments like oral medicine and perio department doing polymorphism of gene for a particular disease, oral diseases. So RFLP is a go, or if you have to learn more about single nucleotide polymorphism, then gene sequencing is the one. RFLP is restricted fragment length polymorphism. Okay. So biomarker based research, this is an area of interest for me. So if somebody is interested to do something on, you want to either identify a biomarker or you want to validate a biomarker which has been reported very recently, or which you have identified from the literature, this is the way you have to go about it. You have to know the sample from which you are going to identify the biomarker. So in case of, let's say, I have worked with oral cancers, if it is going to be oral cancer, then you need to take the oral tissue or the saliva, the nearby sample. And then what method you're going to use? Are you going to use simple bioassays? But now these days, for biomarker identification, they prefer omics analysis, because this gives a clear picture of which molecule is going to be altered. So that the outcome may be a presence or absence or increase or decrease of one or more molecules. Now, you have to compare it with the control and analysis. Why I'm telling this is, most of them, they do design this, but some they don't have a control to compare. Without this, you cannot come up with the biomarker identification. Okay, let's say that you are in the phase of biomarker. You want to do a validation of already reported biomarker. Then you know where the biomarker was identified. So take that sample, but you should know, you're not going to see whether they have reported it. If it is going to be present or not present, or whether it is going to be higher or lower, that you need to measure. So you need to know which tool you have to use, whether enzymatic method or ELISA-based method or flow cytometry-based method or imaging-based method. So that you have to fix. Then again, you have to control with control as well as a gold standard, which is already existing. As in the case of cancer, you know, histochemistry is the gold standard. So you should show that the biomarker which has been identified, it is not necessary that your research should always show a positive mark. It can also have a negative outcome, and that can also be reported. Okay, so you have to show that with the existing method, compared to that, this is far more sensitive and selective. And it is cost efficient, at least you should show that the false positive and false negative is lesser than that of the gold standard. If that can be done, it is the best biomarker. Okay, and I know most of many departments, at least prosto, endodontics, and other departments, few other departments, even Perio, they do dental material based research. So when you are going to do a dental material based research, it can be a graft material or it can be a implant, which is based on steel, or it can be a filler material, any material or a bracket, whatever you want to characterize. You need to know that there are various phases for dental material, and which phase you are going to select. Based on that, you need to approach your research. Say, for example, you want to develop a new dental material, then you should focus on how the synthesis is going to be. And mind you, such things cannot be done by an individual group. It might take toll on you, it will be too exhaustive. So better to do a collaborative research. But you can learn the synthesis also, because they may be knowing the basis of this. They may know how to troubleshoot if any problem occurs. So you can do, but better to collaborate and then do it. So from synthesis to characterization, then test it in vitro model and you can report it. That is one way. Or you can take a already reported material, which has been reported for some other application, or it has been reported in a paper for a basic bone regeneration. That material you can take and you can, but still, even if it is reported, if we are going to prepare it, if it is not bought commercially, ready-madely, if it is not bought, then again you have to do the characterization. Whether the characters which has been reported is similar in the one which I have also prepared, that you need to do. Otherwise, it is not going to be the same. When you are going to prepare in the lab, synthesize in the lab on your own, you need to do the characterization. You may tell, no, madam, this has been already reported. No, it might be reported, but again, when you do, there might be a lot of variation. Because material science is a field, like even a slight change will cause a lot of changes in the morphology and physical characters. Okay, so this you need to know. So let us see what are the different techniques required for dental material based research. Okay. So if I'm going to synthesize it, synthesis type, you have to collaborate with the material scientist. Okay, then you have to choose it. Now, what are the techniques for characterizing this? This I have shown for nanoparticles. This also goes for biomaterials or nanomaterials, which you will be using. Anything, not only for nanoparticles, for all materials. So basically, they will be doing physical, chemical, and biological. Biological, you will be seeing in the next few slides. So physical characterization is for analyzing the size, shape, charge, surface area, morphology, and the release or the degradation, how it is degrading. So how we do this? So if I have to know the morphology and particle size, morphology, surface morphology by SEM, that is surface electron microscopy, TEM gives you the size of the particle. Okay. Then if you are using a particle in solution, then SEM and TEM cannot be used. We have to go for DLS, dynamic light scattering. That will tell the size of the particle in solution, and not only that, it will give the charge, the zeta potential of the material. Again, as important is the viscosity of the material. Some of you might be using hydrogens, membrane-based material and all, and the viscosity, the contact angle, that is the hydrophobicity and hydrophilicity has to be measured. So viscometer is there, and for contact angle measurement, meters are there. So that is there. Contact angle measurements can be done. And BET isotherm is used for measuring the surface area and porosity. If you are going to use some mesoporous materials, say, for example, like bioglass, or if you are going to use mesoporous silica materials, right? Even mesoporous hydroxyapatite has been reported now these days. So if you want to know the pore size there, the BET is the best method. Basically, the morphology of the powder, whether it is amorphous in phase or crystalline in phase, that can be measured by XRD. And how the material is releasing its component. For example, if it is going to be a graft-based or for bone regeneration, how the remineralization is happening. That can be done in vitro itself using a simulated body fluid. It can be SBF, which is like PBS, phosphate buffer saline, having the pH and the mineral component exactly like your blood, or it can be done in salivary, like fluid. So that is also possible. So these are the physical characterization. Then chemical characters, what are the functional groups and what is the characteristics of components, chemical component present in that? This can be done by FTIR, NMR, Raman spectroscopy. So mostly you see spectroscopy is used. Likewise, I told you that the mineral remineralization and demineralization, that is what is more important to you, right? When we talk about using bone graft or filler materials. So how fast it is happening, what are the inorganic ions released during the mineralization, which one is more released? If you want to know that, there is a particular method called as ICPOES, that is inductively coupled plasma spectroscopy. So that is there. Similarly, you can also do the electron arrangement. This is all advanced. They mostly this kind of EDAX is all done by material scientists. Then the surface chemistry and surface analysis can be again done by X-ray, XPS, okay, ion mass spectroscopy and XPS. So this is about how we characterize physical and chemical properties of a material. And importantly, here I have mentioned you about the mechanical strength, because that is more important, right? When we go for hard tissues and even for soft tissues, you need to know the mechanical strength. So that is used, you must be knowing, at least a conservative department must be knowing, UTM is used. Universal Testing Machine is commonly used for measuring mechanical strength. Other techniques are also there, but that is the one which is commonly employed.
Rajashree, can you wind up a little in a few minutes?
Yeah, that's a few more slides, man. That's all.
Sorry.
Okay. So biological validation of biomaterial. This is more important because you need to know it is not causing toxicity. So you have a lot of toxicity assays. Cytotoxicity can be done and genotoxicity can be done. And in vivo also it is tested for preclinical trials. So what are the things that is tested is tissue compatibility is evaluated by histological measures, and they see how far it is causing the foreign body reaction is by measuring the inflammatory cytokines and other degradation particles inside the organism. So that is, then they move on to the clinical trials. So this is about the preclinical trials which you can try for new dental materials. So key aspects of in vitro and in vivo biocompatibility, these are the things which you can perform in vitro also. Like cell material interaction can be studied, osseointegration, osseoinduction, and osseoconduction can be done in vitro as well as in vivo. The bacteriostatic or bactericidal property can be studied because as in case of bioglass, it itself has got an innate antibacterial activity. So that can also be tested in vitro. So probing the cellular response of biomaterials. How do we probe the cellular response? As I have already mentioned, you have the microscopic techniques, or you do the PCR analysis of the genes, or biochemical assays can be performed and analyzed with various flow cytometry or calorimetry assays can be employed. Cell compatibility, I have already discussed about that. So this is the one I was talking about. You have the enzyme mitochondrial reductase, which will change the color of the dye, and when it is when the cell is undergoing cell death. So flow cytometry, this is the picture of that. But the important thing for any material to start with is the hemo compatibility. So how far the material is compatible with the blood without causing adverse reaction or without causing rupture of the RBCs. So you need to see the hemo compatibility. It is just placing your material with the blood and seeing the release of heme and analyzing it with calorimetry or UV spectrophotometry, the simple assay. And also thrombogenicity can be studied, how far it is causing thrombus. This can be done even in vitro. So in vivo models, I think I have already mentioned. So histological analysis can be done to study the changes in the tissue. X-ray radiography can be performed for analyzing the implants and surrounding tissue. Micro-CT is a better one for quantifying the bone regeneration. Not only that, the physical symptoms and health, general clinical aspects has to be monitored. And fluorescence analysis also can be performed for analyzing the bone formation and its progression. So in case of animal model, I think I discussed this already, so I'm not going to it. Depending on what application you are choosing your material, based on that, you choose the animal model. It can be a small model or a medium-sized or large model. So what you want to analyze, based on that, you choose your animal model. So finally, this is about the systematic secondary research. So in the next talk is going to be on that. So I'm not going to touch it, just to keep you aware that if you're going to perform any systematic review or meta-analysis, it is now mandatory to register these also before starting it. So these are the platforms where you can register: Prospero, Research Registry.
Rajashree, for a second, Rajashree, your slide is not moving.
From where?
From actually in vivo, in vitro and in vivo biocompatibility.
No?
No. Is it the same for Dr. Ponnappa? Is it?
Madam,
Are you able to see?
It is still on the aspect of in vitro.
Oh my God. I will stop and again share probably. So now, are you able to see?
Yeah. Yeah. Now, slide show, please.
Yeah.
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 Yeah. Now it's moving.
So I think I'm not going to elaborate on this. So I told you about the different cellular response to material can be studied by all these methods. Then
we were talking about systematic review, I think. Okay.
Yeah. Now the cell compatibility assays, and this also I have discussed, the histological assay, micro-CT, X-ray and all. Then what kind of animal you have to choose. Then this is about the final slide. So you have to register before starting a systematic review, the topic. If once you have chosen the topic for your systematic review or meta-analysis, these are the platform for that: Prospero, Research Registry, InPalsy, and the generic one, old one, is OSF Registry and Protocols. The next speaker I will elaborate, I hope so. Thank you.
Thank you very much. I think I'll take over for 2 minutes before Professor Ponnappan can introduce the next speaker. Thank you very much, Madam Dr. Rajashree. Really, few of the things which I learned myself. The first one is, which I would put across, is for this organ on chip, something really interesting. And I really did not know about this organ on chip. So what I did was, the first question which came to my mind was that, do we have anything for dental organ on a chip? So I thought I'll put that question to you. Do we have anything at present where we can do something on our endodontics specifically, or dental, please?
Yeah, pure dentistry for endodontics, I don't think they have developed an organ on chip. But in case of oral cancer, they have developed oral cancer-based model on organ on chip. And by 3D printing, they have developed spheroid kind of models, in vitro models, which will mimic exactly how the oral cancer tissue is going to be, with all blood vessels and all the fibroblasts, etc. So not for others. Maybe you have to take on, dental fraternity has to take on.
It's something really interesting and something very useful, which is going to be in the field of endodontics, ma'am, madam. I think something which can be worked. The only thing is, how feasible is it to develop it in India? Maybe is something which is, because I just Googled and saw some articles. All the articles are 2020 and 22.
Yes.
One on this organ chip, and they have something on dental. I didn't go through all the articles immediately, but they talked about measuring the biocompatibility. So maybe really fast, quick, but research needs to be done. The other thing is, what I thought was, these tools are extremely necessary. So there are two ways about it. One, you think of a thesis topic, and then you identify the tools which you would require for conducting the study. Sometimes I feel if we know which are the tools which are available with us, we can plan our studies according to that. Now, that's where the question is that, and maybe a suggestion, that is there any list of all these tools being available with specific institutes? Or,
yes.
So,
there are lists in because most of the national funding agencies like TBT and DST, they give the funding, because whatever tools I told about omics or for material characterization, they all come in crores. So no individual universities or even national institutes, they get the funding and buy. So they provide the listing of where it is available, central facilities, and we are allowed to do the characterization in such institution with minimal payments. So even our university, we have a central research facility. So if IITs, they have, everybody has.
So that's the message for a lot of students and researchers, that sometimes we give up a particular research topic thinking that that research tool may not be available with us in our country, the feasibility. But as madam said, there is a central facilities which are on the sites of the funding agencies like DST and DBT. Maybe if we visit them and we contact the coordinators there, maybe we get helped there. I think a really interesting topic, madam. In dental, we rarely touch on these type of topics. So congratulations once more, please.
Thank you, sir.
Very much. Thank you very much, madam. Thank you very much, Arundeep and Dax, please. I think Professor Ponnappan can now take over. Thank you.
Good morning, everyone. Ma'am Rajashree, you've given a very exhaustive description of all the research methodologies. Of course, you have a lot of questions pertaining to the same again. The first question is, what all can be in the in silico models which can be used for dental research?
Yeah, dental research, we can, whatever I have listed, all those techniques can be employed for dentistry also. So for example, a research question is pertaining to development of a drug, docking studies can be done. Or if you want to analyze how a particular molecule, two proteins are interacting, or a bacterial protein is going to adhere to the surface of the caries, or the tooth structure. If you want to study at the molecular level, molecular dynamic simulation tools are available. It's not available in all the institutes, but compared to other characterization, it is easy to procure it. If you are going to spend at least 15 to 20 lakhs, you can get the softwares and the computers super fast, at least fast computers, and we can perform that. So likewise, toxicity studies that can be performed, almost all. So it depends on what kind of question you have. If you want to perform it in silico, it can be done. It can be used. But the thing is, we need to have a computational biologist as well as a bioinformatician to perform it. Not a dentist cannot go, because I'm a basic researcher, wet lab researcher. Even I'm not an expert in doing that. By just following the instruction, we may not do it because a lot of mathematical formulations and physical parameters that has to be optimized, which they will know better. So that is the thing. So you need to have a bioinformatician for that.
Okay, I think you answered the next question also quite well. The next question is pertaining to, are in silico models good for research in MDS? I think your first question answer itself was sorted that out.
Thank you.
The third question is, could you explain the difference between atomic force microscopy and scanning electron microscopy?
Yeah. Say both of them, I think I didn't mention about atomic force microscopy. So scanning electron microscopy, they give the surface morphology, how the surface is, whether it is going to be, what are the peaks and all those things. Even your atomic force microscopy gives you the roughness, but it will give the fine at the atomic level, it will give the surface roughness alone, the roughness of the material. Whereas it will give an overview of your what you call scanning electron microscopy. It will just scan. It will not give you the roughness. It will state these different shapes on the surface of a material. So that's the difference.
Okay.
And at the level of how principal level, how it is working, there is a lot of difference, but I don't think that is going to be useful for the students as of now. Yeah, at the principal also level also how it is working, there is a lot of variation.
And the next question is, can in silico model softwares be used in mRNA estimation? mRNA estimation.
mRNA, yeah, there are tools for analyzing genomics also. Yes, there are tools. I don't know the tools as of now, but there are in silico tools for analyzing DNA and mRNA. There are.
And what's the accuracy of these models for conventional techniques like RT-PCR or ELISA?
No, you cannot substitute the conventional techniques. I have already told that. We cannot do that. We can say, for example, if I want to analyze in the whole of the gene, we have 30,000 genes. Instead of analyzing each and every gene, I can get an idea from the in silico model. Okay, out of this 30,000, this 1,000 or 100 is more prominent or related to this disease. And those 100 you have to test in conventional methods. Experimental wet lab experiment has to be performed. In silico is not a substitute for all that, but it is a accessory. I will tell it is a additional tool, or if I have to say, we wet lab researchers always tell them, oh, yours is like a pickle. It's like a pickle only. You cannot rely on that, but it will give you an idea. It almost takes you near the result. It is not the result, but near the result.
And one last question is, like, you've really put in a lot of data about different methods. So the question 2-3 people have asked is there any particular textbook or an article which compiles all the different tools that you mentioned in your presentation?
Not all. Some of them are in one research article. I can share that with you. Others, I just took it from different sources and then compiled it. So I will share the research articles related to that. No issues. And if you want other research articles for in silico and organ on chip also, that can be done. No issues, sir.
Okay. Thank you for the question and answer session. You really answered most of them really well. I think you'll have a lot of people emailing you later with a few more questions. Thank you, ma'am.
Thank you, sir.
Nice
Thank you so much, Rajashree. Thank you.
Thank you, ma'am. Thank you for the. I should thank you for the opportunity. Thank you.
Thank you, ma'am. Really wonderful.
Shall we continue with the next?
Yes, sir.
Yes, sir.
It's my pleasure to introduce the next speaker, that is Dr. Suneel Kumar Chinni, who is currently working as professor at Narayana Dental College, Guntur in Andhra Pradesh. His area of interest is on clinical research, and incidentally, 3 of his systematic reviews have been published in Journal of Endodontics, in Journal of Indian Prosthodontic Society, and Evidence-Based Dentistry Journal. And that really speaks volumes about the kind of work he's been doing. It's really very nice. And he also received the second best prize in systematic review and meta-analysis competition held in 2019 by Indian Prosthodontic Society. And he's one of the mentors in the Trident Clinical Research Forum, which runs workshop on systematic review and research methodology workshop. So that really speaks volumes about the work which the next speaker, Dr. Suneel, is doing. So welcome, Dr. Suneel.
Good morning, sir.
Morning.
And I would like to thank Team Manva Rachana Dental College and Team SRM for giving me this opportunity. And sir, I'm very, very happy to be part of this thing. And one more thing is Dr. Ponnappa sir is my second BDS teacher. And I'm very happy that sir is chairing my session. That's very wonderful for me. Thank you, sir. So, ma'am, shall I go ahead with my lecture, ma'am?
Yes, please. Yes. You can share the screen.
Good morning, everyone. Ma'am, can I able to see, ma'am?
Yes, we can.
Yes, correct. Able to see. You can do a slide share.
Yeah, I'll continue with my lecture, ma'am.
Could you just full screen it, please? Slideshow.
Slideshow, please.
Sir, is it fine, sir?
No, not yet. Slideshow. Go down onto that icon and do the slideshow, please. Yeah.
Yes. Yes.
Perfect.
Good morning, everyone. This is my institute, Narayana Dental College and Hospital, where I work as a professor in Department of Conservative Dentistry. It's located in Nellore, Andhra Pradesh. And before going to the session, it's more of an interactive session. I'll be happy if postgraduates are answering to my question in the chat box so that I can go to the next slide. If not, it is tough to carry this lecture. So it's more of an interactive session. So first I'll go with the definition of a systematic review. Systematic review is defined as a review of the evidence on a clearly formulated question that uses systematic and exemplic methods to identify, select, and critically appraise relevant primary research and to extract and analyze data from the studies that are included in the review. So basically, you are conducting a review on a particular research question and you are collecting all the articles pertaining to that research question and extracting the data and do an analysis for that comes under systematic review. So that's called as systematic review. So when you look at the hierarchy of evidence, systematic review stands at the top of the evidence. So it is like evidence-based dentistry or evidence-based medicine. Whatever it is written in the systematic reviews, they take it as a guideline for a policy making or for any adaptations in the clinical protocols. They use those systematic reviews conclusions for their research. So when you look at, there are studies like case reports, case series, and case control studies, cohort studies, and randomized clinical trials. On top of that, we have systematic reviews. So basically, systematic review is a filtered information where we are going to analyze the data of all these studies. Either it can be the studies of randomized clinical trials, or it can be from the studies of cohort, or from the case control studies. So we are going to analyze the data in systematic review. So there is a difference between a literature review, systematic review. So basically on a literature review, we summarize a topic that is broad in scope and we don't do any analysis for that. And it is a qualitative one. and sources are might be biased, where we get that can be a bias, and it cannot define what type of studies that can be included. So basically, it is something like a chapter in the textbook. Sometimes they include all the studies, but they don't do any analysis in that. That comes as a literature review or narrative review like that. And when it comes to systematic review, it answers a specific clinical question, like a PICO analysis, and then it defines a specific search strategy where all the included and excluded articles are included. And then it can include a meta-analysis from that also. So we are going to analyze on a particular research question and collect all the articles pertaining to that research question and do a review on that. That becomes a systematic review, which is conducted in a systematic manner. And when it comes to meta-analysis, it looks at studies from systematic review, and its purpose is mainly to combine the similar studies and to pull the data to get a statistically significant result. So when you are conducting an analysis of the reviews included in the systematic review, that becomes a meta-analysis. If you don't do any analysis, that becomes a systematic review. So in the systematic review, we only talk about the qualitative part. So whereas when it comes to the analysis area, that becomes a meta-analysis, we talk about quantitative. What results are there, and we are going to analyze the results again. That becomes a meta-analysis. So not all reviews are systematic review. As I told, the difference between a narrative review or a literature review and a systematic review. Basically, in a narrative review, you have a many questions and there is no search method or no inclusion criteria, and there is no combining of the studies, and it is more prone to random or systematic error, and it provides a conflicting summary. This is basically about a narrative review or a literature review. When it comes to systematic review, you have only one question, and it is exemplary research and can be reproducible, and it includes inclusion criteria, and it combines many study results. That becomes a meta-analysis. So this is the basic thing about a systematic review and where the systematic review stands in evidence of dentistry, and how do you differentiate between a narrative review or a literature review from a systematic review. So this presentation, basically, I have a systematic review for first year postgraduates, what information or what you can take forward from a systematic review, or systematic review general information, that if you're planning to do a systematic review, what general information. So basically the presentation covers this two parts. I'll be happy if which part need to be covered first, if you can answer part 1 or part 2. So I take covering of that one. I hope people are there.
Yes, sir.
Yeah. I'm just looking there. Yeah.
Everybody's there.
Part 1. Yeah. Yes, sir.
Yeah, that is one.
Otherwise, I'll be talking. I don't know where they are, sir. That's the reason.
That is a flip point of online presentations.
Yeah.
You can see the numbers, but the ones, always remember, the ones who are interested would always be there.
Yes, sir. Yes. But still,
So
to me, yeah. Most of the time I see part 1, so I go with part 1 and then with part 2. Since it is for first year postgraduates, if I go to the general information, and they will be forgetting about what they need. So I'll cover with the first part only. So how much helpful is a systematic review for first year postgraduates? So they joined just 6 months back. So if we go to bombard them with a systematic review and then ask them to get this thing, because very new into the department, if you start speaking on a higher level, they might not be understanding that one. So how, as a first year postgraduate, they understand this one. So I'll put forward a scenario 1 where when your guide gave a research question to you and asked you to collect the relevant articles on that research question, what you're going to do? You search a Google database and get the relevant articles, or B, you search PubMed database and get the relevant articles, A and B. And I have one more choice. I have one more choice. Wait. I have one more choice. Wait for a few days. On a lighter note, don't collect any articles. Your guide only will give relevant articles. So which one will you answer for this? Either you go with choice A, choice B, or choice C. Hope nobody gives choice C. Yes. Everyone is answering choice B. Yes, we are going to. Ma'am, Mahalakshmi ma'am has given choice. I hope staff only will tell this was choice C. So the same thing. So wait for a few days. Don't collect any data. Then guide only will give some articles on that, and they will proceed on that. Yeah. Choice B. Okay, fine. Okay. So I have a research question now. So in the previous sessions, many speakers would have talked about the research question and what is FICO, population, intervention, comparison, and outcome. So I'm not going in detail about that, but basically the research question I'm going to talk what is. In this question, I'm talking about what is the success of dental implants with immediate loading versus early or conventional loading of fixed prosthesis. So if I'm going to place a dental implant, if I load it immediately or if I load it delayed, conventional loading of fixed prosthesis, what is the success? This is my research question. As a first year postgraduate, what will you do? So you go, as you said, you go back to the PubMed database and then search the immediate loading and conventional loading. When you search like that, you will get 646 results. So is it possible for you to look into the 646 results and get the articles of those things to read, extract? At least you need 3 minutes or 2 minutes for each article. So is it possible for you to do in that way? Can you answer me? If you're going to type immediate loading and conventional loading, you're going to get 646 results. So you're going to read all those things. No. Okay. So I'll refine my research question saying that immediate loading and conventional loading and systematic review, I'll get 52 results. At least to one point, yes, there are 52 results. Okay. Still more, I can refine it. I can refine my result. I'm looking at prosthesis. So immediate loading and conventional loading and systematic review and fixed prosthesis, I'll get 13 results. In that 13 results, I can take one title which suits more near to my research question and search in that. If it is a systematic review, all the information that is needed for me will get into that. So as a postgraduate, since you are in the beginning in the search of your dissertation thesis topic, you need to search many thesis areas. It is not one thesis topic where we can just take one topic and then go into that. That's not possible. We have to look into at least 5 to 8 areas or 10 areas of thesis topic. And when we are looking into these thesis topic, each time, if you go into each area of 600 results, 200 results, that's not possible. But if you take one research area, one systematic review, you get a lot of information from that. I'll just show you an article in this now. So this is the immediate versus, sir, can you see the screen, sir, the systematic review?
Yes, you can see.
Yeah, okay. So this is the immediate versus early or conventional loading dental implants with fixed prosthesis. And this is a systematic review and meta-analysis of randomized controlled clinical trials. So, sorry. So if at all, I take this article, if at all I take this article, in this article, the author has included, when you look at this, included studies, 49 plus 1, 50 articles were included in this study. 50 articles were included in the study. When you look at the characteristics of the included studies, you have study, which country, what type of study design, and what is the follow-up, and what is the age, and how many patients were seen, and what implant they have used, and what is the loading time. So totally of 50 articles were covered in this systematic review. So now, if you can able to obtain the data, what is needed for you. So for example, I told success. Success is many other things are there in that. When you look at the survival rate or failure rate or marginal bone loss or so many parameters are there to define the success. Which parameter you are discussing on that? You have n number of studies from this, and you can analyze from this one article itself what all research has already been done, what all research has already been done, and the question what you have formulated, whether it has been answered in the systematic review or not. And if at all it is answered, is it necessary for me to stop my research, or can I modify my research question and go ahead with my clinical research? So this is the main advantage of taking a systematic review of a particular research question, because you will come to know what all studies are already being published on that area, and what type of studies is that, and what are the outcomes they have evaluated in that. So when you look at this, this author has mentioned about many outcomes in this article. He has mentioned about marginal mole level change and so many changes he has mentioned. So you can go through one article and find out what all the outcomes that has been published already and what you want to do in your next study. That is one important. That is one advantage of taking the systematic review for your reference. So basically, all the data related to immediate loading versus conventional loading of implants is available in one article, and all possible outcomes related to the system research question is presented in one article, and almost 50 randomized clinical trials were included in the systematic review. And all the related data of these 50 randomized clinical trials were present in one article. And it is an easy way of reference to all the articles. So this is of particularly important for first year postgraduates who is looking at doing a clinical research as their thesis or a paper presentation or a poster presentation. If you have a research question, try to look into the systematic review, and then you will come to know what all research has been published in the literature and how to go forward in the study. So I'll just keep a pass. Any questions are welcome. I'll go to the next one. Any questions in the chat box you can keep related to your thesis. If you have finished any research question you have with you and you have gone with the search strategy, if you have any questions, you can ask me. I'll go to the next part.
I think so. They're saying no questions. So I think probably you could go.
Yeah, yes, sir. No questions. I think I'll go to the next one.
Right.
So the next one is interpretation of a systematic review article. So when you join in the department, maybe your senior, someone will take a presentation of a systematic review in journal club, in the journal club. So look at into the systematic review. How do you interpret a systematic review? So interpretation of a systematic review article. It's basically you're there in the journal club. You just want to see what all areas are there if someone does a systematic review. So when you look at a systematic review, any article will have an introduction, material methods, results, and discussion. And based on the type of the research, they will modify in the material method. If it is an in vitro what type of material methods, and if it is a randomized clinical trial, what type of material methods, it will be defined. But when you come to the systematic review, you have basically a title of the article, research question, and then PICOs, that is population, intervention, comparison, outcome, and study design. And then you have an inclusion criteria, exclusion criteria, and Prisma flow diagram, characteristics of the included studies. And in the results part, we have a risk of bias. And then if you go, the next one in the results part is forest plot diagram, and then discussion and conclusion. These are the main contents of a systematic review article. So most of the article will be under this. You have a title, research question, PICOs, inclusion criteria, exclusion criteria, Prisma flow diagram, characteristics of the included studies, and the risk of bias, forest plot diagram, discussion and conclusion. So I'll just show you an example of a systematic review. Sir, the screen is visible, sir?
Yes, sir, visible.
Yeah, okay. So this is our systematic review which got published in Journal of Endodontics. So effect of preoperative corticosteroids in patients with symptomatic pulpitis on postoperative pain after single visit root canal treatment, a systematic review and meta-analysis. So when you look at this, you have everything like this, Prisma flow diagram, material methods, review, all these things are there, forest plot diagram, and then grade assessment, discussion, and conclusion. Everything will be there in the systematic review. So any systematic review article is there. These are the main contents for that, and it is useful for your clinical research also, or for your clinical applications also. This when you are able to read a systematic review article, because you can gain a lot of knowledge from that, and you can use that knowledge for your clinical practice and as well as your future research. I'll just go with each component of this. I told you title. Title in this article is effect of preoperative corticosteroids in patients with symptomatic pulpitis on postoperative pain after single visit root canal treatment, a systematic review and meta-analysis. This is the title. And the research question, the aim of this systematic review and meta-analysis is to find the effect of preoperative use of corticosteroids on post-endodontic pain in patients with symptomatic pulpitis who underwent root canal treatment in single visit. So we have clearly defined in this, we are going to take only the single visit treatment patients with the preoperative pain and who has administered corticosteroids were only included in this study. So first one is the title and then the research question, and the next one is PICOs. Interpreted the research question in PICOs format. The population what we are looking at is patients with symptomatic pulpitis, and the intervention what we are looking at, whoever has given a preoperative medication with corticosteroids in a single visit root canal treatment. We are going to take those studies, whoever has used intervention with preoperative medication with corticosteroids and done the treatment in single visit root canal treatment. And then they have compared the preoperative medication might be with a placebo or with other drugs and a single visit root canal treatment. And the primary outcome, what we measured was how is the percentage of patient that has reported to the clinically significant reduction in the pain, and is there any pain score between the groups using a validated pain scale. And these outcomes were measured at 6, 12, 24, and 48 hours time period. And secondary outcomes, what we measured was any analgesic medication were taken in the first 48 hours or any adverse effects were reported. All these things were included as a PICOs analysis. And then we have all these things were included as a PICOs analysis for the research question. And only randomized clinical trials who has performed the randomized clinical trials were included in this systematic review. So basically, we are looking at randomized clinical trials which are already published in the literature. Using this research question will be included or were included in this systematic review and carried out the review. So in this, you have a different types of research question. In general, when you talk about the research question, the research question, what I mentioned in the review is about intervention type, but you have a different types of research question. When you are looking at the proportion of the population in newly diagnosed with this problem is called as incident type of research question, or what proportion of population is currently living with this problem is called as prevalence type of research question. And what should be done for this problem? You call it as a therapy related research question. If you're going to detect with early detection of this problem, make a difference in my health, that becomes a screening type of research question. How good is this test at diagnosing this problem? So is there any test which diagnoses this problem? That becomes a diagnostic accuracy test. And what is the likely outcome of this problem? That becomes a prognosis related research question. And will there any negative effects of this intervention? That becomes a harm related one. And what is the cause of this problem? So that is called as etiology related research question. And how can this problem be prevented? That is called as prevention related research question. So basically, the research question in this one, we have taken an interventional one or a therapy related one, but you have a different types of research questions. For example, what is the prevalence of endodontic lesions in type 2 diabetic patients? Then that becomes the prevalence type of research question. And how do you identify the endodontic lesions in diabetic patients? That becomes a diagnostic accuracy one. So the research question can be of any type. It can be an incidence one, or a prevalence one, or a therapy one, or a screening one, or it can be a diagnostic accuracy one, or a prognosis related one, or etiological related one, or prevention related one. So as I come across with the research question, so I've elaborated what are the different types of research questions that are there. When you are looking at your research question, you need to look which research question you are going to address. As previously Dr. Rajashree ma'am also told, it is more important of how good is your research question and what outcome you are looking at. That is very, very, very important. If your research question is already addressing the one which is already present in the literature, that may not add any importance of your review. So when you look at, if you're sending a review to any journal, why the journal need to publish your review? That question you need to ask first. If that journal is getting so many of papers, instead of publishing your paper, what is gaining for them? That is very, very important if you are starting any research. If you are addressing any, I don't say a unique one, but if you're addressing exactly what you are thinking and it is not there properly written in the literature, yes, you can go forward. That is the importance of research question. And when you look at inclusion and exclusion criteria, we have clearly defined in this, we are going to include only the randomized clinical trials and adults with symptomatic pulpitis with or without apical periodontitis. And there should be a comparison of pre and post-operative pain using a validated pain scale, and studies reporting both vital and non-vital teeth, but giving a data separately for vital teeth, and the patients whoever underwent single visit root canal treatment, and then patient who received pre-operative corticosteroid and measured a different time interval, that at least 48 hours time period is there. All those things, whoever has defined in their randomized clinical trial, those articles were included in this systematic review. And it is an important point when you are trying to do a systematic review, you need to clearly mention the inclusion criteria. What type of study design is very, very important if you are addressing or planning to write a systematic review, because study design will have a very important one when you are assessing your results in the systematic review. I'll come back to that in the next part. What is the importance of study designs, what we are including in our systematic review. And the exclusion criteria, what we have used is, it has to be one visit root canal therapy and pulpal pathosis. That means more than one visit root canal therapy, and then pulpal pathosis involving non-vital teeth were excluded. These studies were not included in our systematic review. So these are the different types of study designs that are there: case control, cross-sectional, cohort studies, randomized clinical trials. So these are the different types of study designs. So I have a small question. What type of study designs need to be included in systematic reviews? Is it only cohort studies or case control studies, cross-sectional studies, or randomized clinical trials, or all of them? Sorry, it is D and E. Sorry, it is D and E. I've mentioned it as CC. Sorry, D and E. Please answer in the chat box. What type of study design need to be included in the systematic review? Yes, everyone has mentioned it as randomized clinical trials. That is D option. Yeah, that is D option. Everyone has mentioned it. Before going to the next one, I want to clarify that usually the randomized clinical trials are included in the systematic review, but it need not be that we need to include all the randomized clinical trials in the systematic review. If we are taking a research question that is pertaining to the case control, then we can include all the case control studies and do a systematic review. So if the question is related to the case control, if I feel that what is the risk factor or what is the risk factor associated with this case, if I feel that case control studies are ideally to address this question, then I'll include only the case control studies. And if I feel that my research question is addressed properly by a cohort study, because I cannot expect randomized clinical trials in this research question, and I can only take cohort studies also and do a systematic review. And if at all I'm looking at a prevalent type of studies, and then I can include cross-sectional studies and do a systematic review. You can include either only case control or only cohort studies or only cross-sectional studies or only randomized clinical trials and carry out a systematic review. It need not be only the randomized clinical trials should be included in your systematic review. I have taken an example of my article where I've included randomized clinical trials, but need not be systematic review, need not be only on randomized clinical trial. It can be on case control, cohort, cross-sectional studies also. Okay, so yes, yeah. Any questions? Any questions?
I think there is none in the chat box right now, Sunil. So as soon as we have some, we'll try and take them at that stage.
Yes, sir. Okay, sir. Okay, sir.
They are typing to question which type of study design needs to be. Okay.
Yeah. What are cohort studies? Cohort studies are longitudinal studies where you are addressing one group of patients with treatment. And then articles need to be selected from PubMed. No need to from PubMed, any database. It can be Embase or Google Scholar or PubMed or any other data, Scopus also. Any article can be included from any database you can include in that. Is the clinical relevance of a systematic review reduced if it's not a randomized clinical trial? It's a good question, actually. When you have a research question and you feel many randomized clinical trials are there in that research question, it is better to include only randomized clinical trials. And you feel that there are no research in that particular research question, if you don't get any randomized clinical trials, then yeah, your value of the systematic review doesn't reduce if there are no any randomized clinical trials on that particular research question. You can include cohort studies also and carry on your systematic review. And the average time for a systematic review, it will take somewhere between 1.5 years to 2 to 3 years. Minimum time it will take 1.5 years to 2 to 3 years time. Yeah. Okay. So I'll move forward, sir.
Yes, please.
Yes.
Yes, please.
Yeah. So when you look at any systematic review, they represent it as a Prisma flow diagram that is called as primary reporting items of systematic review and meta-analysis. Prisma stands for primary reporting items of systematic review and meta-analysis, and they follow this diagram, flow diagram, in any systematic review. So they'll show identification, how the identification was done, and how the screening was done, and finally how many articles were eligible, and finally how many articles were included. So when you look at this, included qualitative synthesis is 5 articles. When you come down, quantitative synthesis, it is only 3 articles. So qualitative synthesis means I'm going to tell all these 5 articles how good they are or how poor they are. In the sense, risk of bias, when you take a tool of risk of bias, and if I say this article stands low risk of bias, means that has a very good weightage. And if I say this article has high risk of bias, means that will have a less weightage. So when it comes to the quantitative synthesis, it's called as meta-analysis. In qualitative synthesis alone, if you do, that becomes a systematic review. And if you do a quantitative synthesis, that becomes a meta-analysis. Though that is the difference between a systematic review and meta-analysis. Okay, so and another important one is characteristics of the included studies. I've told you that we have taken 5 articles and we have included the what possible data we can include from the included studies. We made a table, what sample size they have done, what type of tea they have used, and what is the operative diagnosis, and what is the interventional drug they have used, and what route of administration. In that, it is up to the author how to extract the data, and you feel that data is important to the readers. So when you are looking at a systematic review, if you look at this table, characteristics of the included studies, you will get that what all parameters that have been addressed by those authors and the reviewer has presented in the form of a table. So as I told you previously, if you are looking at to do a thesis, if you collect a systematic review, just go into the characteristics of the included studies. You get enormous data of that, what all already has been done. From that data, you can see whether you have planning to do it has already been addressed or not addressed. When you look at the results part, we have given the results what has been done. So sometimes outcome is also measured. So the outcome is very, very important, what we are trying to come out at the end of the study. So if you're planning for your thesis, then you can always see whether these outcome has been already addressed or not by looking at a systematic review. So when it comes to the results part, we have something called as risk of bias. So there are this risk of bias is evaluated by Cochrane risk of bias tool. They have 7 questions, and we need to check in the article whether author has been addressed these 7 questions properly or not. If they address it, we call it as plus. If they don't address it, we are not clear about it, that becomes a question mark. It becomes an unclear. And if they didn't mention it, negative, we give a minus. So based on that, if I get all plus marks, that becomes a low risk of bias study. And if I get only one question mark also, that becomes an unclear risk of bias. Low risk of bias stands a good weightage. That means they have no errors in that article. They followed a good methodology and publish. Even then, published articles, the next question is usually, if it is published also, we need to check the risk of bias. That author has been published, but as a reviewer, you need to check what is the risk of bias of the included studies, what you are checking. That is the main objective or main work as a reviewer for you. So this statement says that all meta-analyses are systematic reviews, but all systematic reviews are not meta-analyses. So if you are doing a meta-analysis, that means you have to do a qualitative analysis and then come to the quantitative analysis. So when there is a data where you can compare the data, then only you will go to the meta-analysis. Okay, but all systematic reviews, but I am able to do a qualitative analysis, but I am not able to compare the data, what has been published. There is so much difference in there. Then I cannot perform a meta-analysis. So that becomes a systematic review alone. So all meta-analyses are systematic reviews, but all systematic reviews are not meta-analysis. So yeah, there's one more question. Is risk of bias tool is same for all type of study designs? Is risk of bias tool is same for all type of study designs? Oh, it's very nice. All first year PGs answered it very well that no. Yes, it is correct. And very glad to see everyone has answered no. Not even one has answered yes. I was expecting yes also, but no, no one has answered yes. Yes. Yes, because you cannot use the same tool for all type of study design, because you have a different tools for each type of study design. So what are those different types of risk of bias tool? You have a Cochrane risk of bias tool that is used for randomized clinical trial alone. And you have a Newcastle whatever scale that can be used for cohort studies and cross-sectional studies. And you have a JBA risk of bias tool. And then JBA risk of bias tool, they have given risk of bias tool for each type of study design. So you have a different tool. You have to take that tool and address your included study article based on this tool, and then tell that how good or what is the risk of bias that article stands. So coming to the next part of the one, that is forest plot diagram. Whenever we see a forest plot diagram, this is how it resembles. You have a diamond shaped one and a small square shaped one and a small square shaped one. I don't go deep into the analysis of forest plot diagram, but I just basically say that how do you analyze that one? I'll just tell. So when you look at this chart, I have a prednisolone which was compared with placebo at 6 hours. That means preoperatively prednisolone was taken and placebo was taken in another group, and postoperative pain was compared at 6 hours. If you look at 3 articles has performed this analysis. That is Al-Khadam article, Dallas-Bed article, and Praveen's article. So these 3 articles have performed this analysis. Now I have included in the forest plot, and this diamond shaped one gives the overall effect. This diamond gives the overall effect. When you look at the center line, onto the left of the center line, we call it as favors prednisolone. On to the right of the center line, we say it as favors placebo. if this diamond is towards the left side, that means the sample is saying that it favors the prednisolone. That means patients who has taken the prednisolone is having less amount of pain at 6-hour group after the treatment when it compared to the placebo. That is what this forest plot diagram is saying. So you have to look at the diamond and you see that where the diamond is lying. It is on the left side or on the right side, and you look at the interpretation what the author has given. The author has given it is favors the prednisolone. And you will look at the small square boxes. This represents the sample size of the individual study. The smaller the box, the lesser the sample size.
I think we have lost connectivity with Dr. Suneel.
Professor Punappan, I think we've lost the connectivity, please. I think the organizers will have to take call on that, please. Dax, are you there, please?
Sorry, sir, just checking on the connections and all.
Okay.
Just give us 2 minutes.
I think something at sir's end.
Yeah, looks like, sir.
All of us are able to connect.
Yeah, we are here. Anji, Anji, Anji.
So we have called sir and he said he'll be joining in a minute.
Sorry, sir, there is some network error. Got disconnected.
No problem, Dr. Sunil. All waiting. Once it's done.
Nice to have you back.
So I've given you the sharing rights name, so please. Yeah.
Yes. I think I stopped at conclusion.
Yes, yes.
Yeah, okay. So some network issues, sir. Sorry for the interruption. So within the limitations of this study and this review, it can be concluded that preoperative administration of single-dose corticosteroids such as prednisolone and dexamethasone in symptomatic pulpitis can reduce the incidence of postoperative pain after single visit root canal therapy in short term and reduce the intake of rescue medication with no incidence of drug-related adverse effects. So it has clearly shown from this systematic review in any symptomatic pulpitis cases, patients, if you are doing a single visit, so you can give a single dose of corticosteroids to reduce the incidence of postoperative pain. So this is the basic thing about interpretation of a systematic review. And most of the time, what is when you are looking at a systematic review, that is fine. When you are planning to do a systematic review, please define your outcomes properly and your inclusion criteria properly so that you can give some evidence to the existing literature. But if you are going to have so many outcomes and so many study designs and so many things, finally you cannot address the question properly. That is where most of the time when you see the proper conclusion won't be there. But if you have a proper research question and you need to go according with the research question and you feel that no study has addressed that research question, then there is a lacuna in the research in that area where many randomized clinical trials need to be addressed on that. That is a important one. So just we have one more small so when you look at this, sir, can you able to see the screen, sir? So I'm just showing a review. Can
Yes,
you able
can
to
see Dr. Sunil?
Yes, sir. Okay. So can you look at this? What is the author is doing in this? Instead of PICO, he has given a PECO. What do you mean by E in this? Instead of intervention, what is the author is looking at? Can anybody in the chat box, instead of I, author is looking at exposure. Exposure is looking. The main title of this article is The Impact of Cardiovascular Disease and Endodontic Outcome, a systematic review of longitudinal study, that is cohort studies. So they are looking at if there is a cardiovascular disease, what is the endodontic outcome in that cardiovascular disease patients. That is what the author is looking at. So we'll go to the risk of bias. Can you tell me what tool the author has used to assess this risk of bias? Can you tell me what tool the author has used for assessing the quality of evidence? You can type in the chat box. You can type it in the chat box also. What is the tool the author has used for assessing the quality of evidence? Anybody? So if you look at the table, there are questions, but the table is in different orientation. You have a type of disease, selection, representativeness, selection of exposure, outcome of was presented not. So they have a different domains in that. and they will answer that one. And you look at this down, good quality means they need to get 6 stars. If it has a fair quality means they'll get 4 to 6 stars. And if they have a poor quality, they'll get 0 to 3 stars. Yes, everybody has answered in the chat box. That is, the author has used Newcastle whatever quality assessment form that is separate for cohort studies. That tool they have used for assessing the quality of evidence, And this is the forest plot diagram. This is the forest plot diagram where it is clearly shows that diamond is favoring towards the control. That means no one is having the disease will have a better outcome than those who are having cardiovascular disease. So the endodontic outcome is good in who are not having cardiovascular disease than who are having cardiovascular disease. That is the outcome is meta-analysis forest plot diagram. So what information we can take from a systematic review for a clinical application? So when you look at the systematic reviews and when you are looking at the textbook chapters also, when one particular area, they'll be writing as per the systematic reviews published on this research area, we can conclude like that. They'll be telling that. So it is better golden standard things means they've already tested that one. If I say 5.2% sodium hypochlorite is a good endodontic irrigant, and based on all the systematic reviews or analysis, they will report in the textbooks that this is good. So in the same way, systematic reviews when done properly on that area and it has been done properly, and it is again waste of time to evaluate those things again and again. For example, if I say anticoagulants are used for cardiovascular disease, it is done. But you cannot again do a randomized clinical trial saying that one group of anticoagulants I'll give another group I don't give anticoagulant, because it's already proven. You cannot do a randomized clinical trial on that again saying that without giving anticoagulants. Either you can give anticoagulant and test another drug. That is a different one. So if it is a proven one and it comes into the clinical application based on the published systematic reviews, then it is based of resources doing it on the same areas again and again. Okay, so information from the systematic review is always used for clinical application. And I'll go to the next part of the presentation, that is systematic reviews, general information. So what is the purpose of doing a systematic review? It is better to date summary of all available evidence and to allow large amounts of data to be assimilated, and those findings can be incorporated into evidence-based healthcare. So that is very important. When you take a systematic review and those findings can be incorporated into evidence-based healthcare. That is the reason of doing a systematic review. And there are university exams where they have a basic methodology. They'll be asking in last final year paper in our university asked a question on systematic review and meta-analysis. They asked a question of systematic review and meta-analysis.
10 sheet है 2350 की हमें चाहिए 2350 मल्टीप्लाई बाय 2 करके.
So even for your theory point of view also it is important that you need to know what okay.
और फिर जो बाकी की जो बचते हैं ना
Participants to please mute their mic. Please mute your mic.
235 225 5875.
Yeah, 4500 multiply by
3 packet.
Okay.
Yes, sir, please go ahead.
Systematic reviews. Yeah, systematic reviews can show which treatment and prevention methods have been proven to work and what remains unknown. That means already proven also it's shown, and we know where is the lacunae in the research. That is also shown by the systematic reviews. And they're very important for pointing out areas where more research is needed. So when they find a lacuna in one area, so they say that you need a more research in that area. That is good for that another people to carry out a randomized clinical trials in that area. And systematic reviews are the basis for what is often called as evidence-based medicine or evidence-based dentistry. So who all get benefits by systematic review? It is by individual doctors and researchers, patients and consumers, and guideline producers and policymakers. Usually all these people, when you look at the policymakers, if they need to implement a policy of drug regime or on anything on a bigger areas, on a epidemiological theme, they always depend on systematic reviews. They conduct a systematic review on that particular drug regimen and get a proper one, and then they use that one, policy makers. That is very, very important for them. So these are the people who get benefit with that, individual doctors and researchers and patients, consumers, and guideline producers and policy makers. Who undertakes systematic review? Independent authors, can be an academician or researcher or clinician, and they can conduct with Cochrane collaboration, largest international organization that conducts systematic reviews, and even they can have a health policy making bodies, professional societies or government, etc., and business people like pharmaceutical companies. These people undertake systematic review. When you look at 2 years back, ICMR announced a proposal for systematic review with a funding of up to 10 lakh rupees. 2 to 3 years back, they have announced on that to send a proposal of doing a systematic review, and they are given the areas of interest also. In these areas of interest, if you send a proposal to us, they give a funding up to 10 lakhs. That's, I mean, talking about 2 years back. So these are all people who are very interested to undertake the systematic reviews. And the Cochrane Library, it is one of the biggest library where systematic reviews are produced or done with high calibration and with high quality of evidence, and it addresses all areas of medicine and dentistry. So when you want, you can look at the Cochrane Library and you can browse by topic wise also. They have a browse in this like this. If you have a research question, you can take that research question and type in the Cochrane Library also. You will get an answer for that, and it is free, and you can have access of this published systematic reviews under them. If you got a publication in Cochrane Library itself, you don't need to publish it in any other journal again, because Cochrane itself is the biggest impact factor journal. Like, it's a big library. So that is the importance of Cochrane. And now recently there is another one called JBA, Jonah Briggs Institution, where they separately conduct Cochrane reviews. So this is the Cochrane database of systematic reviews and the aims and scopes of that. So what are the stages of a systematic review? As I told, I showed you the contents. In the reverse version, we have the same thing. You need to develop a focused research question. I'm again saying that research question plays an important role, and you have to address it properly and define your inclusion and exclusion criteria properly, and then search the literature and then select the studies based on your inclusion criteria, assess the study quality, and then extract the data and analyze the present the results and interpret the results and draw conclusion. And if needed, after few years, you can update your systematic review again. If you have many randomized clinical trials has been published in that area. So it is called as an updating your review. So these are the basic stages for a systematic review. So what authors usually do is they identify the issue and determine the question. Basically, you can generate a research question based on your clinical knowledge. Then whenever you are doing a treatment, when you feel that this area I'm getting a problem. So usually it starts like that. So whether this question has been addressed or not. So you can look into that. When you feel that no research question is not there, try to search again and again. Still you feel that that is not addressed. Yes, you got a good one. You can start writing a systematic review on that. But I say that please go to all databases like PubMed, Cochrane, Scopus, Google Scholar, all the databases, and even in Prospero also, you need to register your Cochrane, systematic review before starting it. So when you enter into the Prospero, you can search the Prospero registry also. You can search the Prospero registry also, where already the registered protocols will be there. Whether any protocol has been registered, if they're registered, you can contact that author also, what stage it is, and you will know whether to change your protocol or not. So all the databases, please search properly. Still you feel that it is not addressed, yes, you can take forward. The same thing if I told you before, plan for the review protocol, search for the studies, sift and select studies, extract data from the studies, assess the quality of the studies, combine the data, and discuss and conclude overall findings, and you get a systematic review. So getting started, so define the research question, clear background, scope, and setting of that, and then research question into the PICO analysis, and specify the inclusion and exclusion criteria. Inclusion and exclusion criteria. So the research question should have these things in mind: defining inclusion and exclusion criteria, searching the literature, selecting the studies, extracting the data, and analyzing and presenting the results. So inclusion criteria can be a combination of population, intervention, comparison, and outcome. The study type also need to be there. Like what study type you are using. Is it a randomized control trial or observational studies, cross-sectional studies, case control studies, and qualitative studies? So basically, this is the one: patient, problem, intervention, comparison, and outcome gives a focused systematic review. Yes, you need to check published as well as unpublished research also. If any dissertation is there, you feel that the dissertation has addressed thesis or dissertation that has addressed your research question properly, but it has not been published, but you have to include that one also in your systematic review, stating that unpublished data. You have to search unpublished data also. But sometimes how it feels that I'm writing a research question, doing a systematic review. I came across that some of my colleagues has done a similar paper presentation I've seen in a conference, but they have not published the data. Yes, I can contact them, get those paper results, and then use it, that one, in your systematic review. If needed, you can utilize it in meta-analysis also, and then publish it. Don't leave unpublished data also. That is also very, very important. It adds weightage to your systematic review. So you need to search comprehensively, search for gray literature, and search for unpublished studies also. That should be there when you are doing a systematic review. What all the recommended databases: PubMed, Embase, Cochrane Central Register of Control Trials, and the interdisciplinary databases like Scopus and Web of Science. All these databases, you need to look into that when you are doing a systematic review. And apart from that, there is a new database, that is JBI. They also do a systematic reviews. You can collaborate with them and do the systematic review with them. So this is the Cochrane Library. So it is free to India, and the national subscription was bought by ICMR between 2007 to 2016. So take home message. So it is clearly framed research question with a proper study design of included studies gives a good systematic review. And I would like to thank to Dr. Mahalakshmi ma'am and Dr. Arundeep sir and Dr. Dax Abraham sir for giving this opportunity to present at this workshop. And like to extend my thanks for judging my session, Dr. Ajay Logani sir and Dr. Ponnappa sir. And this is my contact, email ID and website. And this is our forum where we do a systematic review workshops, interactive workshops. And thank you. And I would like to extend my thanks to Dr. Shubha for helping me this presentation. So session is open for questions. I have some questions. Yeah, how do you avoid repetition of topics in systematic review? You can search the databases and see already published is there, and it is very similar to your outcome. Then better change your research question. How long does it take an average time to complete? As I told, it will take 1.5 years to 2 to 3 years of time to complete a systematic review. Some good statistician with whom we can contact for automata, and you can contact to my mail ID. I'll tell you that one. I have to check that one and tell you. Yeah, this is my mail ID. Mail me. I'll see that and tell you about the statistician. Sir, any questions?
Yeah, there are a couple of them, Dr. Sunil. I'll start with the first one.
Yes, sir.
Is the clinical relevance of systematic review reduced if it is not a randomized clinical trial?
Sir, not reduced, sir. If in that research question, no randomized clinical trial has been published means, then we can do an observational study also as a systematic review. It doesn't reduce, sir. But if you have a randomized clinical trials, include only randomized clinical trials.
Okay, thank you. The next one is, I've seen systematic review of case reports. How do we determine the FICOs in that case?
Sir, in that case, we don't have a comparison, sir. We don't have outcome. In PICOs, we have a problem and intervention only. Actually, I have a case report, systematic review on case reports also on COVID-19 related one. There, they don't have a comparator and outcome based. They have a case and report that one. That's all, sir. You can't have any comparator and outcome in that, sir.
Okay. I hope that answers the quite a relevant question that was.
Yeah. Yes, sir.
The third one you've already answered, that is how long does it take to complete? We'll skip that. The next one is, please explain what is the gray. Just a minute.
Gray literature, sir.
Yeah, gray literature.
Sir, gray literature is unpublished data, sir. So it can be a paper presentation or a poster presentation or any dissertations. You need to go and search in the university websites or anything like that and then get on that, sir. Gray literature is mostly an unpublished data is called as gray literature.
Unpublished, does it mean a specific kind of standard journal or
Sir, unpublished means it is not published in the journal, sir. It can be presented as a paper presentation at any conference or poster presentation or their thesis or a dissertation. Those becomes an unpublished data, sir.
Cool. And the one more is, what are the minimum number of articles required to conduct a systematic review?
Sir, even with one review also we can do a systematic review, but see that no article has been there in that proper search. Even with one article also we can do a systematic review. Or else a minimum of 2 they say that, but I've seen one couple of systematic reviews what they have done with one review only, one article only.
And in the chat box, one more person has asked a question: how to avoid repetition of topics in a systematic review?
So to avoid repetition, we need to search the databases and see that a similar research question has been published in the systematic review or registered in the Prospero. If at all it is similar, then try to change our research question looking at a different outcomes. That's the one way of avoiding repetitions.
I suppose it's a last question. All the questions are there. In fact, in the chat, there are a lot of them have written that it's a very informative lecture, and they were asking to reshare the link for the feedback form.
Sure, right.
Has done nicely, you've reviewed everything. Very systematically, you've done a review of the talk. Very nice.
Thank you, sir.
Thank you for sharing your presentation, and it was good to interact with Dr. Ajay Logani and get in touch with Dr. Arundeep Singh sir and Dr. Dax and Dr. Vakul, and thanks to Mahalakshmi ma'am for this opportunity. Thank you all once again.
Sunil, can you stop sharing your screen, please?
Yeah, yes, ma'am. Yes, ma'am.
Thank you.
Yes, ma'am.
Thank you.
Yeah.
Okay, I think we've had really 5 days of exhaustive learning, and thank you, Dr. Chinni, for such a wonderful presentation. And thank you, the chairpersons for the day, Dr. Punappa and my friend Ajay. And we are much beyond scheduled time, but we have one more, like they say, icing on the cake in store for all the participants. So we had planned a workshop in a mode that, we'll have some kind of a discussion. So under Professor Vidya and Dr. Kritika, we're going to do a kind of a hands-on type of a workshop on synopsis writing. And initially we planned it to be for limited attendance, but with the kind of overwhelming response we got, we said, okay, let's open it to all. So I know it's already 2 hours and probably another hour and a half of good discussion, but it'll be really worth the while for everybody to attend. So I would hand it over to Dr. Mahalakshmi to start this workshop on synopsis writing. And I'm sure all of us are going to be really, really benefited from it. And let's make it a success. So all those who want to leave are obviously can leave for now. So this is not related with the certification of the program, but it is for everybody's interest to be in there and, get the best out of this. So over to you, Dr. Mahalakshmi.
Thank you, Dr. Arundeep. And thank you, dear participants, for the overwhelming response that you have shown to us for the past 5 days. And we have been consistently having participants every single day logging in at, sharp at 9 o'clock, 100, at least a minimum of 100. And Dr. Arundeep wanted to start it only when there were 100 participants. And you all made sure that it happened. And we started on time, and it was a phenomenal experience that we have had. So this finale is just a small introduction for the first year postgraduate students to how to write a synopsis. Because most of the universities have a different format in which they have to submit a synopsis. Some universities do not have. And if you present it in a proper structured manner, it makes a lot of difference, and it presents that you are really interested in doing it properly. So this small workshop, I know it is not enough to not enough time to go through each one with each one of you as a hands-on. So we have just taken up 2 articles, 2 dissertations of ours that is already been published. So we take you through the process of our synopsis presentation and through the journey. And Dr. Vidya and Dr. Kritika will explain in detail about one aspect of and one in vitro study and one in vivo study that we have completed and published, and how we did the synopsis. And we'll take you through the process today. Over to you, Vidya.
Yes, ma'am. Ma'am, I want to share my screen.
Ma'am, just giving you the right.
Okay.
Yes.
Kritika ma'am, please check.
Yes, ma'am. I'm able to share.
I'll just give to Vidya.
Yeah. Good morning, all my dear teachers and students. I thank ma'am and Manav Rachna Dental College for this opportunity. So myself and Kritika. Screens very frequently, so there might be a lag when you see my screen on your computer or laptop. workshop, so kindly bear with
Excuse me,
it
Kritika ma'am.
because I have to show the points in the PowerPoint and how it was actually written in a synopsis.
Excuse
What
me, ma'am.
kind of corrections you
Vidya,
might
I think you're
receive
not
from
audible very much.
IRB.
Kritika ma'am is not audible. There's some problem with Kritika ma'am's network.
No, this is Vidya, so it's Vidya's whose network. Vidya?
Okay.
So in this workshop, we'll just see the.
Maybe you can turn off your video and try, ma'am. Vidya, ma'am.
Suchita. Ma'am.
Just give us a few seconds, she'll do it.
Yes, ma'am.
Sorry for the break, but we'll be back. in a
Sir, that was just a technical glitch, so I'm very sorry about that.
Vidya ma'am is back, yeah.
Ma'am, I've given you the co-host rights. Please check once.
Yes, ma'am, I'm able to share my screen.
Yes, ma'am. We are able to hear you.
Yeah. So I'm sorry about that. So coming to the nuances of synopsis writing. Before getting into a dissertation, we'll just decide on a topic, right? So how I'm going to show a few slides about how we decided upon a topic for the study. And before that, I would like to show the published article.
Oh.
This is an article published in European Endodontic Journal. So in this workshop, you're going to see how the idea of this article came up, and from a synopsis, how it got published into an article. So this is what we'll be showing you. So we just started with a small idea as to compare the properties of AH plus sealer with the same sealer which is modified with a herbal medicament, an anti-inflammatory herbal medicament. Why did we want to modify the sealer? So there should be some drawback for AH plus, right? So that drawback was what we tried to focus. So how do we focus on the drawback? We have to get into the literature, read about AH plus, what are the problems associated with AH plus, and then find out a solution to resolve that. This is how we came up with this study. So the main problem with AH plus was on extrusion, it will produce an inflammatory reaction. So we have an article to prove this. So that
your slide is stuck. It's not moving.
Yes, no, sir. I will keep.
It's supposed to be like
changing.
that. Okay.
Yes, sir. I'll keep changing, sir. I'll be showing a few articles in between, so I'll keep changing.
Right.
Yeah. So this was the article which gave us a spark that AH plus induces inflammation in a cell line. So this is where we got the idea that something is wrong with AH plus. So we can modify it. And the same author, Kim et al., had published the study in 2013, giving an insight into a new herbal medicament called as pakimic acid. It is a fungal derivative product widely used in Chinese herbal medicine, but it's not explored in India at all. So this product, we decided if added to AH plus, what will happen? That was our idea, because it had a proven anti-inflammatory property. It had a proven anti-inflammatory property, right? And then the previous year, we had published another article saying that pakimic acid can be added to a root canal, and the root canal sealers showed reduced cytotoxicity. So these two articles were the nidus for us to begin this presentation.
Vidya ma'am, I think the slide is not moving. It's a Hello, Kritika. Kritika, are you there?
Actually, Kritika ma'am is there, but
Ma'am, just a second, ma'am. Just altering the technical glitch. Just
Yeah. Okay. No problem. No issues. Take your time. No problem at all.
Are you able to share the screen?
Yes, ma'am.
Okay. Share your whole screen. Don't share only the laptop.
Okay, Preeti's article. Are you able to see my screen? Okay. And I'm back again. I'm sorry, ma'am. So this is where this was the study which was published by us. So I'm going to run through the process of how the study started with the synopsis. So before beginning this study, how we got the idea. That's what I was telling when the network busted. So this was the idea which we got from Kim et al.'s article published in 2013, where he showed that AH plus does some damage to the periapical tissues when it's extruded beyond the apex. So that was the lacunae of the material. That was a disadvantage of the material. This disadvantage was overcome by the addition of a traditional Chinese medicine, which was pakimic acid, which is not available in India. Even now, it's not available in India. So this author showed that addition of pakimic acid to AH plus is capable of reducing the inflammation which was produced by AH plus using the cell line. So we had an idea about addition of something to AH plus can control inflammation. There was following this article, we published another study in the Journal of Endodontics by adding pakimic acid to different root canal sealers, and we saw whether this pakimic acid was able to reduce the cytotoxicity of the sealers. To our surprise, the pakimic acid was able to reduce the cytotoxicity of AH plus drastically. But at this time, we never tested what this addition will do to the physical properties or the sealing ability of the original sealer. So there was a lacunae in the literature about the physical properties and the sealing ability of the addition of pakimic acid to AH+. So this was the nidus for us to decide on a dissertation topic for the student. So coming back to my presentation, we formulated a tentative topic to compare the properties of AH plus resin sealer to the same sealer modified with an anti-inflammatory herbal medicament, pakimic acid. And we wanted to do it as an in vitro study because basically we are testing the properties. So I need to find the right keywords to formulate my topic. As the eminent speakers had already spoken, the keywords chosen based on the mesh terms given in PubMed, NLM, was AH plus pakimic acid. Physicochemical phenomena or physicochemical process is the actual keyword. Root canal sealer or resin-based sealer was another keyword. So we should make sure while formulating a title that all these keywords are incorporated. So if you look at our tentative title, we can see that we have used the term physicochemical. We have used the term apical sealing ability, which is another keyword. We've used the term AH plus. Generally, AH plus is a commercial name. we have been advised not to use commercial names in topics, but AH plus is such a widely used epoxy resin-based sealer that it has entered the mesh NLM database. So AH plus as a keyword is acceptable. There are many properties which can be assessed. When you say a new material we are going to formulate, there are many properties. For example, if it's only physical, we can say flow, working time, setting time, film thickness, radio opacity. Chemical properties, solubility. Biological properties, cytotoxicity, anti-inflammatory properties, sealing ability, micro leakage. Here we should exert caution before deciding on how we are going to progress the study. I cannot make the student to do all the properties for a dissertation. So we zeroed in on a few properties and we came out with a topic like this. This was the final topic, comparative evaluation. I'm going to compare the original AH plus sealer with a modified version. So the word comparative is very meaningful here. Comparative evaluation of physico-chemical. I'm doing some physical properties and one solubility as a chemical property. So I coined the term as physico-chemical properties. And among all the biological properties, I've chosen only apical sealing ability for the student because it is impossible financial wise, feasibility wise to do everything for a dissertation. We've not used the term AH plus in the heading because we wanted to use a generic name. AH plus belongs to the journal of a resin-based sealer. So we decided we will go with a generic name in the title. So finally, the title was changed after many discussions with our staff and our HOD ma'am. It was changed to comparative evaluation of the properties and sealing ability of a resin-based sealer. So this is how the presentation slide to an IRB, the first slide, should look like from the student perspective. I'll again stop sharing and show you how a synopsis format would really look like. The original synopsis format, which is given by SRM. I'll share my desktop. So this is the synopsis format which is given at our SRM Dental College. If you see, the first few columns are to share the vital data of the student when you're admitted, what is your course and all that. Here comes from number 5 onwards, the title of your study. Number 8 is the most important column in a synopsis where you give your best to explain to the IRB people why this study has to be approved. What is the novelty in the study? Why the student has to be allowed to carry out this research? So we'll focus more on what all can come under this justification pertaining to the topic which I am talking now. Again, coming back to the presentation. These 3 aspects should be the top priority in my justification. So first, I'll talk about the background. I have to tell the reviewers what has been done so far and what is missing. So coming to the background, first I have to make the reviewer understand the importance of a sealer. So where does the importance of a sealer come? The success of the root canal treatment is directly proportional to how well you debride and how well you obturate, how well you seal the canal. So we all know without a sealer, it cannot be achieved. So you need to refer articles, review the literature and find out relevant latest articles stressing the importance of a sealer and its ability to achieve a good sealing ability in the canals. So this will form your background. After that, I should move on and tell why is the study required. So why is the study required? As we already saw, there is some problem with AH plus which we want to rectify. That is why the study is required. So you will explain the IRB, what happens if the sealer is extruded, how a cytotoxic reaction can produce an inflammatory reaction, how this inflammatory reaction can delay wound healing. So now I'm addressing the problem. Earlier slide, I addressed why is a sealer important. Now I'm telling them what happens if a sealer gets extruded, what damage it could cause, And in the next slide, I'm going to tell how I'm trying to address the problem. So there are a variety of anti-inflammatory agents available, right? I have to tell the IRB why I chose pakimic acid now. So you have to give a brief introduction about what are all the various studies on anti-inflammatory agents available, and then tell them why pakimic acid. 2 reasons pakimic acid is very effective. It's a very effective anti-inflammatory agent. And the second reason, it is not yet explored. The properties of pakimic acid modified sealer, there is not even a single study which is explored. Has it been added to the sealer already? That question will come from IRB. For that, we have our own article which was published in JOE where we showed that it can be added to the sealer and it has been shown to reduce cytotoxicity. So this much of information you have to give in the background and justification of the study. This is very short. Whatever I've shown in the PowerPoint is very short. And again, I'm going to come out of the PowerPoint and show the synopsis which we had submitted. This is how it will look. Okay. So there goes the heading of the synopsis. I'm zooming it. You can see the heading written there. And this is where point number 6, why is the study required? Brief justification. But it's a very elaborate justification that we have given. What are all the studies previously published? I have shown how AH plus is cytotoxic. So at the end of each finding, you will give a reference, relevant literature reference, which will be checked by the IRB people. So I've shown that literature shows that antioxidant and anti-inflammatory agents can give protection, and one such material is pakimic acid. We have shown Kim et al.'s article here, what he did in that study, and we have shown how our own article number 4 reference has shown reduction in cytotoxicity. And then I'm proposing a hypothesis that addition of pakimic acid could reduce inflammation, or it could improve, or it could damage the properties of the sealer. So I have to frame a hypothesis now. So the hypothesis will be addition of pakimic acid to AH plus will not affect the physicochemical properties or the apical sealing ability of the sealer. So here I derive an aim. This is the aim of the study. So coming back to the PowerPoint, here comes our, the same thing which I've spoken, why it is required. So why the physicochemical properties? I'm focusing on physicochemical properties because when we add a new compound to an already existing sealer, what happens? The original formulation is altered. So how will we know when it will set? How much of working time will it give? Will it dissolve when it's exposed to the fluids in the oral cavity? Or will it set or will it not set at all? All these things we don't know because it is something new which we are doing. So the physical properties have to be assessed. That is the importance of deciding on the physical properties. And only if it sets, it can seal, and it should not shrink when it seals. It should not be soluble in the periapical fluid. So sealing ability evaluation also becomes important. So this is how the aim was coined. The aim of the study was to evaluate. If you see the title, we have only wrote physicochemical properties. But if you see the aim, we are zeroing in more specifically on what properties are tested: flow, setting time, film thickness, solubility, radio opacity, and sealing ability. So your aim should be very elaborate. It should convey to the reviewer what you're trying to test. The aim should convey what you're trying to test. Remember that. But when you come to objectives, it's totally a different ballgame. An aim can be very crisp, but an objective has to be very elaborate. Here you have to tell what you're testing and how you're testing. That is very important. Remember these questions, what and how completes the objectives. So this was the objective which we first submitted to IRB. We thought we'll write it very short, the first point. But the IRB gave a comment that your objectives are too vague. Elaborate on the objectives. So we finally came with such elaborate objectives where every parameter shows that I'm testing AH plus modified with pakimic acid with AH plus under in vitro conditions. I'm testing setting time, I'm setting film thickness, I'm testing the solubility, radio opacity, everything in comparison between AH plus and AH plus modified pakimic acid. But if you look at point number 6, it is different. Okay, the testings of working time, setting time and all that. I've not mentioned which instrument I'm using to test. So there is something missing in that objective. But if you see point number 6, it is more clear. I'm telling them I'm going to test the apical sealing ability of H plus and the modified sealer using a fluid filtration method. I'm very specific in this. So why I have not added the instruments in the rest of the 5 columns is because, for example, if I'm telling I'm testing solubility of AH plus, there is no such instrument as such to test. It's a chemical process, right? Similarly, radio opacity can be mentioned. We can mention it was done using a densitometer, whereas the working time, setting time was measured using Gilmore apparatus. That could have been mentioned. So still there is a lacunae in this aim, in these objectives. A more complete objective, I'll give another example. This is a different study. Kindly don't get confused. We are still traveling with H plus and facimic acid, but this is a different study. This article was published in European Endodontic Journal. So look at the objectives here. I'm very specific. I'm telling them I'm using a standard plate method, an agar diffusion method I'm using to test the antimicrobial activity. This objective is perfect. It shows that I'm quantifying the antimicrobial activity as well as I'm comparing the antimicrobial activity of 4 different medicaments. Against which microorganism I have mentioned it's E. faecalis, in which substrate I've mentioned it as human root dentine, under which condition, in vitro condition, using what? Agar diffusion test, or otherwise called a standard plate method. If you look at objective number 3, 2 and 3, they are going to be done under a confocal laser scanning microscope to evaluate the depth of penetration of these medicaments. So this is a very good example of how a complete objective should be. Again, this study is published in Journal of Oral Biology and Craniofacial Research, a prestigious journal run by the Craniofacial Society. In this study, we try to analyze what happens when the dentine is remineralized with various agents. How does composite bond to this remineralized dentine? So what will be the objectives? What tool will I use? I'm going to use a scanning electron microscope to analyze the remineralized patterns of dentine. So I'm very specific. I'm specifically telling the reviewer, I'm going to see the acid etched patterns. I'm going to see the remineralization in the dentine. And I'm going to use a scanning electron microscope for that. The second objective says that the microsphere bond strength testing will be done under a universal testing mission. So your objective should tell what you're doing and how you're going to do. So coming back to our original aim, this will be the aim of the study. As we said, we are going to test the physicochemical properties and the sealing ability. Now, moving on from aim and objectives, we should move on to the study design. The study design is very critical. Most of the questions after aim and objectives will arise in this part of the synopsis. It is very difficult to convince the IRB about the study design, how we validated the study design, what sample size we require, how we validated the sample size, how the samples will be collected, what are those samples, how they will be stored. Just see the number of questions that might arise from this part of the synopsis, right? So first, coming to the study design, I have to clearly tell them 2 types of studies are involved. One is going to test the physical properties, one is going to test the sealing ability. So think, physical properties do not require natural teeth, right? They do not require as such a sample size calculation also. They do not require any scrutiny from the ethical committee also. But when I'm going to test the apical sealing ability, I'll be using natural teeth. Anything which is collected from a living tissue, the ethical committee becomes awake. They become alert. They keep asking me, how are you going to collect the teeth? So I have to tell them, I'll collect the teeth after getting consent approval from the patient. So I have to explain the patient that the extracted teeth will be used for research purpose. You will not claim any rights to the research results or the purpose of the research paper. So we get a disclaimer consent form signed from every patient from whom we are extracting the teeth. I'll show you a sample of the consent form also. So again, coming to these parameters, the source of sample, I've mentioned clearly that the first parameter will use only the sealer samples and the second parameter will use teeth. So when we use natural teeth, then this comes up, sample size calculation. Now the statistician enters the scene, right? We have to give him certain data so that he gives a robust sample size calculation, which when verified by IRB does not get us into any problem. So while giving data to the statistician, two things you have to see. If the study is already done using another material, you can choose that article as a key article and take the mean and standard deviation of the groups from that article and give that data to statistician. So number one, you can use your key article provided a very similar study using slightly altered materials are done. But in this case, it was not the case here because we never had a similar article before. So how do I give data to the statistician? Another technique is you do a pilot study with a small sample size of 8 to 12, and then you give this data to the statistician. This was considered for our study. But the problem was I couldn't do a pilot study when I submitted the synopsis because if you see the apical sealing ability, it needs a fluid filtration model. We never had a fluid filtration model when the student entered barely 6 months into the course. We never constituted a fluid filtration model. We had a plan of constructing as the study progressed, right? So I cannot do a pilot study for apical sealing ability. So what is the other alternative? I can select a fluid filtration article, which has done, which has used similar root samples and done a fluid filtration study. This is how we came upon an article published in 2008 where fluid filtration using AH plus itself was done to evaluate sealing ability compared to various other resin sealers. So that was taken as a key article for sample size calculation. So if you see the sample size calculation, there is the last line shows actual power 0.9. So the power of the study was robust. A 90% power was set before proceeding with the sample size. So the statistician will give you a graph like this, where the x-axis shows the power of the study and the y-axis shows the sample size. Coming back to the graph, you can see the graph has very less power when the sample size was, total sample size was 10. It had only 75% power. The power kept on increasing to 90 and above 90 when I had a total sample size of 24. So considering this data, we selected 8 teeth for each group so that the total sample size came to 24. So this is the process involved.
To add up here to Vidya's calculation of sample size. Now, when you do a pilot study, the samples that you have used, the values that you have used for your samples can also be included in your main study. So the advantage is that when you do a pilot study, it's not necessary that those values will not be considered later. So the number of, accordingly, if you do a sample, say suppose a pilot study of 8, then in your, if your sample size now is 24, then you can consider this 8 also among the 24. So you need to do only another 16. Go ahead with it. Sorry.
Thank you, ma'am. So this is how we derived at the sample size value for this study. Now, as I said, I'm going to use natural teeth. So I have to tell the IRB, where will I get these natural teeth from? See, there has been various legal issues also where students go to mortuary to get teeth and all that. So the IRB is concerned about the safety of the students. IRB is like a big boss which is watching over your research so that your research goes smoothly, right? So I have to tell the IRB, I'll take teeth which are extracted in the oral and maxillofacial surgery department of SRM Dental College, and I will store the teeth in 0.1% thymol until it is used. So every detail has to be given in this study design, sample size calculation, method of sampling, which is random sampling, of course, and how the samples will be stored. And coming to the inclusion and exclusion criteria, which are very critical for the in vivo study, which will later be discussed by Dr. Kritika. So for me, it was quite simple. I'll just be taking premolars, but to test the sealing ability in a fluid filtration model, I should focus on one point, that the root has to be straight. So that is why my inclusion criteria had only one point. It should be a premolar with relatively straight roots. So in the exclusion criteria, I will obviously avoid roots with excessive canal curvature or multiple canals or a fractured teeth, or if the apex is too wide, all these will definitely be excluded. Here again, I'm stressing on the importance of consent form from patients participating in the study. So now I'll just show you a sample of consent form. This is how the consent form looks, where the patient will sign that he has been informed that the tooth has been extracted as part of the treatment procedure and it will be used for research, and he is wholeheartedly giving his extracted tooth for research, and he will not raise any issues later in the future. The same consent form should be there in the vernacular language of your state, which will be shown by Kritika also again. So now methodology, the third most extensive part is the methodology. From methodology onwards, we'll shift back to the synopsis format. In methodology, there are three important steps. One is I should tell them how the sealers should be prepared. Preparation of AH+ will follow the manufacturer's instructions. But when I'm adding AH+ and pakimic acid, I should first tell them how much of pakimic acid should be added to AH+. So we ran a short pilot study by mixing little pakimic acid to AH+, and we froze the concentration, which did not affect the setting time of the sealer. So with that pilot study in the background, we chose a 1:1 ratio of AH+ and pakimic acid, gave a satisfactory setting time and working time, which was the basic parameter which is expected of any sealer. So a short pilot study of setting and working time helped us arrive at this proportion. And the second thing we have to tell, how will I test the physical property? So there are certain standards proposed by ISO for every dental material that we use. So the standard for a root canal sealer goes with the document number 6876, and it was updated in 2012. There are frequent updates which you're supposed to check at the ISO website. So according to this ISO 6876 2012 standardized document, the samples were prepared, which I'll be showing in detail in the synopsis. And the apical sealing ability was done using a fluid filtration model. So again, coming back to the synopsis. Yeah. I'll start, we stopped with aim by showing the aim in the synopsis. So I'll just take you through the review of literature. If you notice here, I have given importance by writing Kim et al. Articles first, and then our own study, Arun et al.'s article published in JOE, and then the Lee et al. Study, who proved that this pakimic acid can counteract the stress produced by hydrogen peroxide, another strong evidence of its anti-inflammatory action. And I'm showing my key article, which I used to calculate sample size, Bulliget et al. 2008. And I'm showing another article which shows that the properties of a sealer can be tested using ISO standards. So the review of literature is again strong. The IRB can check these articles anytime to check whether our claims are valid enough. The objectives, as suggested by the IRB, we've changed. Now, the materials and methods section, if you see, every material, it has to have a manufacturer name, the commercial name, manufacturer name, the city in which it is manufactured, the country in which it is manufactured. For example, AH+, which is manufactured by Dentsply from Constance, Germany, and pakimic acid from Wuhan, China, like that. For every material, we have to give all these details. Note that I have not given the batch number here because at the time of submitting the synopsis, it is not expected. Whereas when you submit your destination, you have to tell them from which batch each material given in the list is used. Not only that, every material that goes into the study, EDTA, hypochlorite, Gatapaka, the glass plate, whatever mold that I manufactured was done by Bandari Metals in Chennai. K files, pro taper files, every smallest instrument has to be mentioned with its manufacturer name, the city of manufacture, and the country. Now I'm going, as I said, there are various parameters which we are testing. So there are several equipments which are going to come into play. So now the IRB will have an idea whether this study is feasible or not. If you do not know which equipment to test your study on, the IRB will not sanction your synopsis. So you have to convince the IRB that I have enough equipments and it is feasible for me to go and work there and get things done. So Gilmore, optical density meter for radio opacity, all these were very much available in Chennai itself, where the student indebted to do the study. So after giving the details of the instruments and the equipments which is required, we are going on to the sample size calculation. As you can see, I have given the sample size here, what type of teeth I want, which are the teeth I'm going to exclude. All these are part of synopsis. Here comes the protocol which is to be followed. Please remember that when the reviewer reads your study protocol, he should be able to visualize the entire procedure. So close your eyes and visualize how we are going to mix the sealer. For example, when you say I'm going to mix pakimic acid, I should know how much to mix. Such intricate details should be there. The flow property alone runs through so much, right? So every detail, one glass plate will be taken, a drop of the material will be placed, another glass plate will be placed, 100 grams of load will be placed. The load should stay there for 10 minutes. Every detail is there in the ISO specification document. The same specification was followed, but please make sure you don't copy verbatim from any document. Make sure you understand and put it down in your own words. For the second parameter also, there is a standard size of the specimen which has to be made 10 mm into 2 mm. So the student made a jig to make sure that this dimension is satisfied by the jig. So every detail, again, 100 gram of mass should be placed, and 3 times this experiment should be repeated. So an average of 3 measurements will be taken as the setting time of that sample. Then film thickness also goes by ISO standards. The solubility also goes by ISO standards. Radio opacity again needs a specialized jig. An aluminum step wedge is required. So all these were synthesized by the student. Coming to sealing ability, the student came up with this prototype. Okay, this fluid filtration apparatus was prepared by us. It was prepared by the student. So this prototype shows that the tooth will be attached at one point. The tooth will be attached at one end of the model. There will be a pressurized oxygen cylinder at the other end. A micro syringe will be used to introduce an air bubble, and the movement of the air bubble will be calculated as the fluid flow through the sample. So we have to check that this setup is converted to a model, and the model should be validated. How can we validate? You keep an intact tooth and make sure that the air bubble does not move in spite of opening the oxygen cylinder. That means that the system is airtight and it is ready to check. So all that was done by the student. And then coming to the last part of the synopsis, you should give adequate references for the reviewers to go back and check. At the end of the synopsis, you should also write what kind of a statistical analysis will be done after you get the results of the study. So your statistician will help you in that. He will tell whether according to the distribution of data as parametric or non-parametric, we'll use ANOVA or student t-test. This we cannot write. You need to, when you calculate the sample size, the statistician will tell you what kind of a data at the end he might be using for such studies. And there is something called Gantt chart, which is nothing but the timeline which you're supposed to tell the IRB. I will collect the samples for 2 months. I'll do a literature review for 6 months. And then I'll procure the materials for the month. I'll do the physical property testing for 1 month, sealing ability testing for 1 month, interpret the results for 1 month, finish off the dissertation. within the stipulated period of 18 months. The last part of the synopsis is the financial outlay. So you have to tell the IRB how you're going to manage the financial part, how much of money you'll be shelling out. So this should also have a split up. How much is going in for the material, how much for the jig, how much for the model, and how much will be your total amount. With this, the components of the synopsis are coming to an end, but the review of literature part, I just wanted to show you how an advanced search bar looks. So when we first started searching with the term anti-inflammatory alone, you could see 7 lakh articles popped up. When we tried to search a general term root canal sealer, 3,000 articles. Not possible for the student to go through all these. So we narrowed down and came to epoxy resin sealer because H+ is epoxy resin sealer. The search narrowed down to 1,000 plus articles. And then I tried to see how much of articles are there regarding pakimic acid. Very less, just 130. Then we started making search builders. So we started with epoxy sealer and studies based on anti-inflammatory properties of epoxy sealers. Again, narrowed down to 127 articles. Studies which assess the anti-inflammatory property of pakimic acid were only 21 articles. If I build the search term with all the 3 articles, epoxy sealer, pakimic acid, and anti-inflammatory property, it only came up to just 2 articles. And those 2 articles were Kim et al. Articles and our own article, which was published in JOE. So this is how we should narrow down our review of literature and select a key article and show it to the IRB people. And then this is the mesh NLM site. Here you should type your mesh term. For example, I said AH+ is a mesh term. As you type, it shows that AH+ comes briefly under the category of epoxy resin based sealer. Like that, every mesh term can be searched. You can put pakimic acid and know whether it's a mesh term or not before adding it in your title. Pakimic acid is there here, right, as a mesh term in front of you. So, and then you need to follow certain guidelines to conduct in vitro studies published by Kritika Datta et al. As CRIS guidelines. We need to check this document also to make sure that we are handling the extracted teeth in a proper manner. Coming back to the presentation, there's another timeline which can also be given, whatever we saw in the synopsis. This is another timeline, but a Gantt chart is more explanatory to the IRB people. Finally, the IRB will ask, what are you trying to prove by the study? So you need to write 2-3 lines about the outcome of the study. This study might enable developing a product that might reduce the inflammation in the periapex, even if it's extruded, right? All these should be given to the IRB. And finally, this is how the published article looked. So as we write the synopsis, it goes through a rigorous correction by our HOD ma'am. And this is how it looks, full of red lines. It shows how much of mistakes we do as we write the synopsis. So ma'am keeps asking, this is irrelevant. This is not needed. Change this, change that. You've not addressed this in the methodology. Where is it? So we need to make sure that every detail goes into the synopsis to avoid these red boxes from your HOD and your guide. That's all, ma'am. Thank you. I really thank the organizers for this opportunity and ma'am especially.
Okay, thank you, Vidya. Now we have seen the in vitro studies being done. So we'll just see the in vivo part, how it has to be addressed. So over to you, Kritika.
Good afternoon, one and all. A goal without a plan is just a wish. So there are dreamers, there are planners, and the planners are the ones who make their dream come true. So have a concrete plan, and that concrete plan is your synopsis. So once your synopsis is properly formulated, then your thesis is going to be published in a well-reputed journal. First, you need to understand these basic terminologies when you design your study. the difference between an in vitro study, ex vivo study, and in vivo analysis. The key difference is in vitro, you do it outside a living organism. Example, it could be a material characterization study or a laboratory-based study. Whereas ex vivo is done outside of a living organism. That is, you take a tissue sample from a living organism, culture it in the external environment, and mimic the natural oral environment in external conditions. In vivo, you do the study inside a whole living organism. So this needs to be defined prior to any start of study. So there are three basic questions that you need to answer when you formulate your synopsis. One is, why do you want to do the study? Second is, where, on whom, and how you're going to do the study, intend to do your study. And third is, what is the outcome? This will delineate all the clutter and gain clarity and help you formulate a good synopsis. The first impression is the best impression. So that first impression is gained by the title of your study. The title of the study should be concise and informative, accurate, unambiguous, specific, and complete. It should be in a professional language, and you should avoid any abbreviation, especially the rarely used ones. It should succinctly describe what was investigated, what was measured, and the sample, that is the measurements where it was taken from. So this is how my synopsis started. My first-hand experience under my own HOD ma'am, Dr. Mahalakshmi ma'am, where we thought of the concept of comparing two different regenerative protocols using induced blood clot technique and scaffold implantation technique in young immature permanent teeth with the 18-month follow-up. An in vivo study was the proposed title. And after scrutinization by the IRB, where they had suggested that we can use an increased sample size with just one technique, which was the scaffold implantation technique, which proved to be better than the blood clot induced technique. So the title changed to Evaluation of Regenerative Potential of Non-Vital Immature Permanent Maxillary Central Incisors using scaffold implantation technique with a 1-year follow-up, a clinical trial. Later for the publication, we had modified the title further with a follow-up of 2 years we had published, where the title was Prospective Cohort Study of Regenerative Potential of Non-Vital Immature Permanent Maxillary Central Incisors using platelet-rich fibrin scaffold. So the next step is the background of the thesis. This is the backbone of your synopsis. So you have to introduce the topic first. A thorough literature search must be done to know about the topic and also reveal what is not known about the topic, and thus in turn will lead to the aim. So the need for the study should be elaborately justified. You should be able to explain the problem in your background of the study, describe your approach or advancement or the change in the experimentation modality that you're going to use, mention the existing solution, that is, what are the commercially currently available materials. You'll have to tell about what are the materials available, what are the limitations of those materials, and why the study is taken up. So in my study, my synopsis, it included the etiology. The etiology here was trauma in young children, and the sequelae following it was the immature tooth, incomplete root maturogenesis, which was explained. And traditionally, the protocol that was followed for the treatment of these immature teeth was apexification. So immature teeth basically led to thin dentinal walls, and the root length was shortened. Incomplete root maturation was there, and the apical diameter was greater than 1.1 millimeter. So with these currently available materials, that is MTA and calcium hydroxide, the apexification attempted did not allow complete root maturogenesis to happen. So this lacunae in literature helped us to determine what was the protocol that needs to be followed for the treatment for these immature teeth, where regenerative endodontics was gaining popularity in dentistry. So with regenerative endodontics gaining this momentum, and there were only limited literature evidence, that is in the form of case reports, we thought of publishing an observational study on regenerative endodontics using scaffold implantation technique, that is platelet-rich fibrin versus scaffold. So always remember, whenever you give your background evidence, you should quote the appropriate references. I would now like to show you how the referencing is done. So first, the keywords that I used was platelet-rich fibrin, which was one of the scaffold implantation technique method. That is the material that was used for scaffold implantation, which revealed a search of 2,631 articles. In general, regeneration revealed an article search in PubMed of 3,73,858 articles. And when I entered the keyword immature teeth, it was 1,823 articles. And when I combined all these three together, it gave me a result of 29 articles, which helped me further proceed with my study. The aim is the goal of my study. So the first proposed aim when we proposed to compare two techniques was to compare the regenerative potential of immature permanent teeth with necrotic pulp using scaffold implantation technique and induced blood clot technique with a follow-up of every 3 months for a period of 18 months. Later, it was then modified to assess the regenerative potential of traumatized non-vital immature permanent maxillary central incisor teeth using scaffold implantation technique with a follow-up of every 3 months for a period of 1 year. And this was taken up further for a period of 2 years. With a follow-up of 2 years, we had published. The objectives of the study is the next step that we need to look into. These are the stepping stones to reach the goal. So for any in vivo study, there would be a primary goal, a secondary goal, and a tertiary goal. We had followed the AE guidelines where the follow-up included the clinical and radiographic examination where no pain, swelling, or sinus tract was noted between the first and second appointments. Resolution of the apical radiolucency was observed. Increased width of the root walls was seen. Increased root length and positive pulp vitality test response was also assessed. So these goals need to be assessed for any in vivo study. So our study based on regenerative endodontics, we had taken the AE guidelines. The next step is the study design. So there are various types of studies. First in hierarchy is the randomized control trial, following which you have the cohort study, a prospective observational study, case control study, cross-sectional study, case reports or case series, and also you have the animal studies and ex vivo studies. So the difference being between the cross-sectional study and the longitudinal study is that when the data is collected at a single point of time, it's called a cross-sectional study. On the other hand, when it's repeatedly collected over a period of time, it's called a longitudinal study. In the case control and cohort study, the difference being when you trace from the effect of a disease to its cause, you call it as case control study. And it is always done in a retrospective manner, whereas the cohort study can be either a prospective or a retrospective study, where we start from the cause of a disease and we trace all the effects that follow it. So this is a pictorial representation of how a prospective and a retrospective study works. That is, from today, if I start a study and the disease is present today, the further sequelae that happens and its effects are being observed and noted, that's called a prospective study. Whereas I retrace back and determine the etiology of the disease, that's called a retrospective study. So these are some of the key components that you need to keep in mind whenever you write your synopsis for an in vivo study. The first key component is the inclusion and exclusion criteria. So we followed, as I said, we followed the AE guidelines. The inclusion criteria included patients with age group of 9 to 25 years with apical diameter greater than 1.1 millimeter, the teeth without or with periapical pathosis with a PAI score 2, teeth with pain, swelling, sinus opening, patients not allergic to medicaments and antibiotics necessary to complete the procedure, ASA 1 or 2. So this is how specific you need to be when you choose your patients. The patients following only this inclusion criteria will be selected for the study. As ma'am mentioned, Vidya ma'am mentioned the source of sample. In vivo study, humans or animals will be the source of a sample. So in humans, you'll have to mention it as patients reporting to the respective department of that particular institution with that particular pathological condition will be included as the source of sample. For example, in my synopsis, it was mentioned as patients reporting to the Department of Conservative Dentistry and Endodontics, so to the Department of SRM Dental College, Ramapuram, with immature permanent teeth and diagnosed as non-vital, that is pulp necrosis with immature, non-vital immature permanent teeth were included for the study. The sample size calculation was done based on our key article published by Bill Kaller et al., which was 39. So we also increased the sample size from 39 to include the 10 and 20% dropouts, which need to be kept in mind, because as it is a follow-up study, we'll have to include these dropout numbers as well. And the statistical analysis needs to be coordinated with your statistician, and the proposed statistical analysis needs to be mentioned. An information sheet should be provided to every patient explaining the process, what procedure is going to be carried out on the patient, and a consent form should be provided, where this written consent form should be signed by the participant, that is your patient, after thorough reviewing it by them. So there's also another process that needs to be taken into consideration before you start your synopsis, that is the CTRI. The Clinical Trial Registry of India, in this website, you'll have to register your clinical trial. This is again, like your synopsis, you will have to provide every detail from the investigator's name, their address, the contact person for your study, the inclusion, exclusion criteria, the outcomes, and a brief summary. So this needs to be done, as this only will be provided. This number needs to be provided when you publish your article. Mine was a prospective observational study, but as I also mentioned, it's a randomized controlled trial. That is the first in hierarchy. So in that, we need to do two procedures. One is randomization and the other is blinding. Randomization, there are various types of randomization. One is simple randomization, the other is block randomization, stratified and simple stratified randomization. The most commonly used one is the computer generated randomization, where we enter the patient data and the computer randomly assigns it into two groups, one being the control group and the other is the investigational group. And by this, you can avoid any bias. The process of blinding, it's basically of three forms. One is the single blind technique. The second is the double blind. And there's another form which is called as a triple blind. So you need to make sure which form of blinding you're going to choose. In single blind technique, the patient is unaware of the treatment process. That is, which either you're going to use the experimental agent or the control agent, the patient would not be aware. Only the doctor is aware, that is the dentist will be aware. Whereas in double blinded, both the dentist and the patient are unaware of the treatment protocol that is being followed. That is, either it's the control group agent or the experimental group agent is not known by the dentist as well as the patient. It's a third operator who gives the agent, and this is called as a double blinded study. Whereas in triple blinded, the participant, physician and the statistician, all three are unaware whether it's a experimental agent or the control agent. This is a study published in our department by Dr. Charanya, Dr. Vijay Amritraj sir, and our HOD, Dr. Mahalakshmi ma'am, on the postoperative analgesia of 2% lignocaine with or without magnesium sulfate for inferior alveolar nerve block in symptomatic mandibular molars. It was, as you can see here, it is mentioned in the title itself that it's a randomized double-blind controlled clinical trial. A 20% dropout was also included when the sample size was calculated. And as you can see here, a block randomization was done. And the blinding was a double-blinded study. The evaluator who recorded the pre and post-operative parameters were blinded to the anesthetic solution used, and the endodontist who performed the treatment also was blinded. These are some of the guidelines that is available with every particular reputed journal, that is, be it IEJ or JCD. So with all the reputed journals, there are particular guidelines that you need to follow. So prior to the start of your synopsis writing, if you could look into these guidelines, you will be able to effectively formulate your synopsis based on these guidelines, which will facilitate your publication as well. The methodology needs to be very specific. You need to include every little detail as possible. So what did you do? These questions need to be answered. What did you do and what do they need to know? What did you do is the researchers in your field need to reproduce every finding of yours when they test in their laboratory site or in their patient cases. So this will help you build on your research, and repeatability is one important factor that you need to allow your readers to know when they're reading your methodology. What do they need to know is who or what will be used in the study. That is, you should include your sample or the participant details, the materials where they were purchased, how will you conduct your study, that is your methodology and techniques should be given in every little detail as possible. You should discuss the specific conditions and control. How will you analyze your data, that is what quantification method is going to be used or software that is going to be used must be mentioned. And also the statistical test, you should consult your statistician and write your proposed statistical test. So these are some of the tips that you can take. Describe how the problem will be studied. Include the detailed information to allow the repetition. Do not describe previously published procedure, but cite them clearly. That is, give credits to the author and just cite them. You don't have to repeatedly put that as well in your synopsis. Identify the equipment and material. That is, feasibility is one key quotient that you need to look into prior to the start of your synopsis. Use proper notations, including chemical formula and symbols. Don't forget to present the controls used. Finally, the outcome of the study. This should be explained in such a way, your work, how well it's going to advance the present state of knowledge. Clearly define your view, create an impact in the scientific field. So this should be kept in mind when your outcome of the study is being proposed. The outcome, for example, in my thesis synopsis was a clinical trial will be a boon to the literature and it will make a paradigm shift in the treatment protocol of non-vital immature permanent teeth, which can be achieved in routine clinical practice. Avoid any scientific misconduct. Do not fabricate your data or give any false information. Avoid any plagiarism or salami slicing. Copying word to word is bad. Paraphrasing, text recycling, or self-plagiarism, try to avoid these scientific misconducts, as your thesis will only be taken up and rephrased for your publication, and this could lead to the retraction of your article. So do not copy another person's study ditto, which will only lead to scientific misconducts and a black mark in your profile. Keep reviewing as many times as possible. Even you're fed up, please keep reviewing, as this only will lead to the progress and make it a better proposal in the form that you can carry out your thesis in a more effective way. So keep reviewing as many times as possible. And this is my thesis, and I'm very, very grateful to my HOD ma'am, Dr. Mahalakshmi ma'am, Dr. Sujata ma'am, and all faculty members of the Department of Conservative Dentistry and Endodontics, SRM Dental College, for helping me publish in my dream journal, that is Nature Scientific Reports, with an impact factor of 4.379. So I would like to conclude by saying that when you're long gone, your scientific legacy is in large part the literature you left behind and the impact it represents. Thank you.
Thank you. And I think we can now have any questions that the students have. If you have any queries, you can even raise your hand and we'll unmute you and you can ask questions.
I'm just wanted to say very impressed with the way how the presentation was done by Vidya ma'am and Kritika, and especially I was impressed with the way how that the synopsis was approval is going on.
Thank you, sir.
So students, this is a time to interact. If you have anything vis-a-vis what you are planning for next for your synopsis, so you have really who's and who of dentistry here to answer your queries. We've had such beautiful interaction all these 5 days, so let's do it better now. This is really time to come out with whatever doubts you have, any questions. You can raise your hand and we'll unmute you also. Let's let it be more interactive.
Then I can assume that everybody understood it very well. So they don't have any questions to ask.
Yes.
We'll take it like that.
Or they've not been listening.
Or the other way is that they have not understood anything at all. So that cannot be after 5 days.
There's a question from Murli sir. Sir's asking, any minimum timeline in place to check outcome of a new material or procedure in prospective cohort? Yeah, Kritika, you can read the question there. It's there on the side.
Yes.
Yeah. Okay. Any minimum timeline in place to check outcome of a new material or procedure in prospective cohort?
Sir, in a prospective cohort, mine was a prospective cohort study, sir. So we had followed the AE guidelines wherein 6 to 8 months to delineate any pain and swelling was the primary outcome or primary goal of our study. So as indicated in the AE guidelines, 6 to 8 months is the minimum time period that we look into. So be it any other material, for example, be it an intracanal medicament, at least 6 months we need to assess how is the periapical healing, or in terms of a newer material that is being placed. So within 6 to 8 months is what we're looking at. So that could be the minimum timeline.
Okay, and to add to that, it all depends upon what study you're doing. If it's endo-related, then you need a minimum of, say, how long can you consider the endodontic treatment as success? It's a minimum of 2 years, actually. So technically speaking, it is 2 years. But when you present it as a synopsis for your dissertation, you do not have 2 years time. So it will be, for the synopsis purpose, we can actually give a 6 months to 1 year. But if you have to publish it, you keep working on it, go through the follow-up procedures till your, by the time you write your thesis, it will be another 6 months by the time you write the article. So you'll have 6 more months for your follow-up. So technically, anywhere 1 year and above is safe for publication purpose. So Murli sir is the only person who's been very attentive, I think.
Good, Murli sir. Nice.
I'll just give it to you. I think we can wind up the session for now.
Yes, ma'am. They're just requesting everyone to join back at 1:30 for the validatory. The same link only will be used. I think
at 1:30. Yeah.
Yeah, 1:30, right? I think that because we're just contacting everyone, whoever can join in, we'll just see. We'll just so as to get a grand.
The participants who are here also are requested. We are close to 100, so we request join back for the valedictory. Since we have had a wonderful 5-day session, we wanted to make it really professional, so we will end up end this program with a proper valedictory.
Grand finale. Grand finale. Yeah, the time for the valedictory is 1:30 p.m. Yeah.
1:15, 1:30.
So आप बताओ 1:15 भी कर सकते हैं. We are all ready actually.
Yeah, I think we can do it 1:15.
ठीक है, 1:15. Okay. Mahalakshmi, 1:15 is okay, right?
Yeah, it's fine. Absolutely.
Yeah.
Thank you.
Okay. Right.
Thanks. You've told Ravi sir to join?
No. Ma'am, could you please ask Ravi sir?
He has gone to Katangattur right now. So I'll just see if he's on he's available, then he will join. If he's on the way, he can join it to his mobile. I'll just find out.
இல்ல இல்ல சார், ஆக்சுவலா நம்ம முடிஞ்சா நீங்க இங்க கட் பண்ணணும். நான் தான் சொன்னேன், இங்க கட் பண்ணணும். ஃபர்ஸ்ட் அதுதான் இது பண்ணுவோம். மேம் கேட்போம், அப்படி இல்லைன்னா கொடுத்து விடலாம். சரி, அது அந்த ஒரு இதுதான்
So should we get it on?
Another 2 minutes, maybe.
Okay.
Ma'am is there? I've not been able to get in touch with the rake last minute.
Ma'am was from coming from Kritika's, I think. But ma'am, are you there?
I'm there.
Yeah, ma'am is here only. Yeah, take it. Okay. Yes, sir. So, ma'am, 2 minutes. We're starting it. Huh?
Sure, no problem.
Is Ravi sir joining, ma'am?
No, he's actually in Katagoto right now, and he's in a meeting. So he messaged me. He didn't pick my call. So he messaged me. He's in a meeting. He'll call me back. So in case he's able to come, then I'll tell him.
Okay.
I just messaged him that if possible, you can please join. So he said you can go ahead, but I don't think he'll be able to come.
We could share the link with sir, no?
He has the link already.
He has a link. Okay, good. Take it.
अरे कर लो यार शुरू, कितना वेट कराओगे?
आप अकेले आए हो ना सर, अभी बाकी लोगों की वेट करें. It was last minute decision. 2 minutes more. I think if the coordinators can confirm that the chairpersons have joined, we can start.
So many of them are saying that they are busy and they would try to. I think so.
We should start and.
Yeah, I think we should start it, sir. Let's start it, sir.
Sure. So, the Dr. Mahalakshmi said, this was a small seed which has really grown into a big tree, and we've had really 5 days of exhaustive, participative learning. And with various, we've experimented with everything. Really, we've experimented with polls, we've experimented with hands-on workshop, and it's been a learning for all of us. And the really high point of this whole workshop was that we had such wonderful interaction among students and the presenters and the faculty, and each of the sessions, I think, had nearly 15 to 20 minutes of question answer sessions, and time was not an issue because we are using this virtual infrastructure, which could be, would have an unlimited use. So like Dr. Ajay was saying, that this pandemic has, yes, caused us lots of misery, but it has also given us tools to connect people from all parts of the world on a single platform for the benefit of the listeners. So I think that was the learning, and that those are the tools that we've used to make this workshop a success. And of course, the real success is the participation of all the delegates who have so well participated in this workshop. I would like to also announce that we have been so encouraged, me and Dr. Mahalakshmi have been so encouraged by the response, that we've decided that we'll have this as an annual event. So the next should be happening immediately after the current batch of MDS joins. So they are giving the exam now. So probably once they join, so we'll come up with a date, but there will be an annual collaborative event between SRM and Manav Rachna to share and for the benefit of the first year students. And I would also be proposing it in the IES meeting, that we would have IES with us as an association and make it an annual IES program also. So it is said that thank you really is, if you have to just say one word, then thank you is the word to say. So this validatory function is for all of us, from the speakers to the organizing team to the chairpersons, co-chairpersons, all who have supported our endeavor and made it a huge success. So over to you, Dax, to have this validatory function on. And my sincere thanks from the bottom of my heart to all of people who have made this a success. Thank you very much.
Thank you, sir, for these wonderful words. And really, as sir has already spoken about it, that I mean, there are so many takeaway points, so many key points, how rigorous the research process is, how we can actually, get good research from people who are actually doing good work in the country. Pan-India attendance, great support from SRM college. I mean, it was, we really come to know where we are lacking also. IAS always is ready to support us at any point. And it's also great news, sir, that it's going to be a collaborative event and it's going to be an annual event. It's really good because there are so many things that we need to learn still. I still feel that we are still, in the learning phase, that learning curve is there. So great work. And I thoroughly enjoyed this personally for 5 days. I really enjoyed it. That 9 to 11 is something from the first day we were looking forward to it. And it was, and like how ma'am very clearly said that this is the time that you have your seminars in the department. If all colleges, they're conducting this. So I think this was a meaningful seminar that we had. It was really a meaningful seminar. Everyone learned a lot. And ma'am, I was truly impressed at the way how Vidya ma'am, I don't know, it's still going on in my mind again. The synopsis correction that you were doing, that is exactly how you do it in the journals also, how the correction comes, you have the red tags coming in over there and this, and I mean, that's truly professional. That shows what level, you guys are at. I mean, it's really fantastic, man. Thank you so much. Thank you so much. Now I just simmer
Dr.
Ma'am,
Mahalakshmi, if you could please say a few words.
Yeah,
before we
Mahalakshmi, ma'am,
start.
please.
No, I think Dr. Arundeep and Dr. Dax have, compiled and told what we have felt over the 5 days. I'm sure, as Dr. Arundeep was saying, we'll have it as an annual affair and make sure that every year we bring out more and more information, or we'll better it. Definitely, we are going to better it with good inputs from eminent speakers. That much assurance we can give you. And yes, as Dr. Dax said, I am a stickler for perfection, but I'm also learning a lot, and I have learned a lot during this 5 days. And I have to mainly thank the participants for coming in large numbers and encouraging us and the speakers that to give our best. So I hope this has been an learning experience for you all, and I sincerely hope that these 5 days will help you to select a proper topic, to do a proper study and publish it in a very good journal. That is our ultimate goal. And I want more and more publications from India, and people should realize, around the world should realize, that we are not somebody who can be taken lightly. So the research that is happening in India is something which they have to look forward to, and they have to come back to us and tell us that, yeah, you be the leader and we'll follow you. That is our ultimate goal. Okay, so thank you so much.
I would request Dr. Sanjay also to chip in and few words from Sanjay. No 150-word restriction this time. So,
I think Dr. Mahalakshmi is driving right now. Look like.
But I'm not driving, so no problem.
Okay.
She drove the whole conference.
You know, I really would like to congratulate Team Manav Rachna and SRM for this wonderful program. But I would like to add something that in my experience, I have seen when you make these programs, annual program, you look forward to improving it. And in the process of improving it, you complicate it more. So please make sure that when you go ahead next year, you improve and simplify it more instead of complicating it. That's the only input I would like to give. And my best wishes to Dax, and Arundeep and Mahalakshmi ma'am. Thank you very much.
Thank you, Sanjay. Yeah, you're right. The idea is we'll have new audience every year. So that's the idea behind making it an annual program. And yes, we'll try and improve. And yes, obviously not make it more complex for sure. It'll be for everybody. And so I think we can go ahead. And people are asking, when will the video link be up? I think by early next week, the video link of this whole workshop will be up on YouTube. And so that it can be utilized by people who are interested in watching it. And let's go on with the validatory function.
Yes, sir. So I just want to put on record, a huge round of applause to Sucheta and the PGs. They have done a fantastic job. I mean, the way how they organized it till Sunday, especially Sunday was the one which I liked it the most, sir. The various meetings that were done, the groups that were created. I mean, it went on flawlessly. From then, I don't know if they did not do it. I don't think this workshop would have actually come to the stage of what we are sitting at. Really, so a huge round of applause, and they're waiting for a huge gift from your side also.
Party on.
Thank you so much, sir. Thank you so much.
Yes, it is always that people in the front are always projected, but the people in the front are supported by so many people at the back. Yes. It is almost 10 times more than what we are projected out. So the hats off to the entire team of Manav Rachna. That's the reason why I took up this workshop, saying that I have not done anything at all this time. So let me at least do this workshops for you all. And thank you, Meena, for joining in.
Hello, ma'am.
I think you represent all our moderators and speakers for today.
And SRM, thank you so much.
Thank you.
Right, Simar. I think we can take it forward now. Yeah.
Kritika, you can begin.
If all the chairpersons and speakers, whoever are present, if they can put on the video, it'd be very nice. So we can have them all together on the screen. I know short notice, but we just thought that it'll be incomplete without valedictory. Thank you, Rajashree ma'am. Thank you, Malti ma'am.
Just a small technical glitch, sir. The network, they're just coming back online.
Yes, sir. Just 2 seconds.
Thank you. Thank you.
Thank you, Chinni sir. Sorry for the slight delay. We'll be on in a minute. Yeah, there you are.
I'm so sorry for the technical glitch, sir. Am I audible?
Yes, Kritika. Yeah.
It is said that end of one chapter is just the beginning of another. So with this, we come to the culmination of the 5-day research methodology workshop. We would express our sincere gratitude to all the speakers and all the chairpersons for their valuable contribution in imparting knowledge, which is so valuable to all the students here. We would like to start by presenting a token of appreciation to Dr. Vivek Hegde, sir, President IES.
In absentia, we've not been able to contact him, but yes, thank you, Vivek. Thanks a lot.
Not to miss, we would like to acknowledge the presence and valuable contribution of Sanjay Miglani, sir, Secretary General, Indian Embryonic Society. Thank you so much, sir. Moving on, we would like to acknowledge Mahalakshmi ma'am, Head of Department of Conservative Dentistry and Endodontics, SRM Dental College. Thank you so much for contributing, ma'am. We would also like to express our gratitude to K. Ravi sir, Dean, SRM Dental College.
The clapping is sounding like disturbance.
Thank you, sir. Without your unrelenting support, this event wouldn't have been possible. Thank you so much. Moving on to the speakers of the first day, we have Dr. Bhagavad Gita ma'am.
First one, no?
Thank you so much, ma'am, for such valuable tips on how to conduct a research. Following this, we would like to acknowledge the contribution of Ashish Kumar sir. Thank you so much, sir, for all your valuable contributions. Moving on to the chairpersons for day one, we have Sonali Taneja ma'am. Thank you, ma'am, for chairing the first session of day one. Along with Sonali Taneja ma'am, we have Meena ma'am. Thank you for your valuable contribution. Moving on to day two, we have Subha Anirudhan ma'am.
Yes, sir.
We would also like to, sorry. Sorry for the technical glitch again, ma'am. We would also like to express our gratitude to Vivek Agarwal, sir. Thank you so much, sir. We would also like to extend our gratitude to Dr. Srinivasan, sir. Moving on to the chairpersons for day two, we would like to express our gratitude to Ekta Chaudhary, ma'am. Thank you so much, ma'am. Uma Sudhakar, ma'am. Thank you, ma'am. We would also like to express our gratitude to Dr. Rohit Kochar, sir, and Dr. Mamta Kaushik, ma'am. And we would also like to express our gratitude to Dr. K.S. Banga, sir, and Sangeeta Talwar, ma'am. Thank you so much, ma'am. Moving on to day three, we would like to present a token of appreciation to our speakers for day three. Dr. Ruchika Runta, ma'am, along with the chairpersons for day three, Dr. Dolly Patel, ma'am, and Dr. Tarun Kumar, sir. Thank you so much for your valuable contribution. We would also like to thank Dr. P. 11L, ma'am. Thank you, ma'am. We would like to present a token of appreciation to Dr. Malati, ma'am.
along with the chairpersons, Dr. Vanita Nikhil ma'am and Dr. Mandeep Garewal sir. We would also like to express our gratitude to Puneet Batra sir. Moving on to the last day, that was today, with very exciting sessions yet again. We would like to express our gratitude to Dr. P. Rajashree ma'am, along with Dr. Ajay Logani sir and Dr. K.C. Panappa sir for chairing the session for today. We would also like to express our gratitude to Dr. Suneel Kumar Chinni sir to end the 5-day workshop so well. Thank you so much, sir.
Thank you all, and really obliged by your presence, and looking forward to yet another session of this program for the coming batch, mid of July or August. So thank you, everybody, and it was really nice having all of you here. Thank you. Bottom of my heart.
Thank you and Jai Hind.
Can we have a screenshot, everybody? Somebody can take a screenshot of this. Banga sir, if you can switch on your, thank you, sir.
Thank you so much, sir.
Thank you, man. Bye. Bye, man. Thank you.